over 21 collaborative models, making a new high. However, compared to global leaders in AI, Sri Lanka is still developing ecosystem. Challenges such as limited infrastructure, a lack of policy, and underinvestment in research and development have slowed the pace of adoption. Nonetheless, a growing number of tech startups and academic initiatives are beginning to explore AI, suggesting that there is a budding potential for AI-driven innovation. Sri Lanka has long been poised to become a leader in the South Asian tech landscape, and recent developments in digital transformation and national AI strategy are promising steps in that direction. However, economic development through technological innovation doesn't happen in a vacuum. We must integrate research in fields like public policy, ethics, economics, and technology to maximize the potential benefits. For instance, AI can play a transformative role in policymaking by improving decision-making process, enabling data-driven governance, and enhancing transparency. However, AI must be embedded into systems in a way that considers that ethical implications such as bias in algorithms and data privacy concerns so that the benefits are shared equitably. This is where interdisciplinary research becomes critical. We need policymakers, IT professionals, sociologists, and ethicists to work together to design systems that are not only technologically advanced, but also fair and inclusive. In Sri Lanka, sectors such as agriculture, tourism, education, healthcare, and public transport stand to gain significantly from AI-driven innovation. The question isn't just how we develop the technology, but how we create policies that enable these innovations to flourish. Because already as a country, we have all this technology, but we don't know how to use it to the right market. Research from various fields, such as economics to understand financial viability, public policy to implement regulation, and sociology to gauge public acceptance, must come together to shape this AI revolution. The National AI Strategy plans to establish essential foundations to enhance the country's position in the global AI landscape in alignment with the Digital Strategy 2030, which focuses on broader digitization efforts in the economy. And the stakes are high. AI has the potential to increase efficiency, reduce cost, and create new jobs across sectors. But without sound research-backed policies, we risk exacerbating inequalities and leaving vulnerable population behind. This is the balance we must strike as researchers, and this is why multidisciplinary approaches are essential. And I have to mention most of this presentation is AI generated. As researchers, we stand at the forefront of shaping this future. I encourage you to embrace opportunities to collaborate across disciplines. Whether your focus is on IT, economic, biology, environmental sciences, or the arts, your research has the power to contribute to economic development in ways that you may still need to realize. The essence of economic development lies in problem solving, and the more perspective we bring to the table, the better equipped we are to tackle complex challenges. For instance, research in climate change should not be only approached from an environmental science perspective, but should also involve economists to measure impact, urban planners to rethink infrastructure, and technologists to design innovations that mitigate adverse effects. In tech, we encourage computer engineers to learn behavioral science and psychology as a modern trend. It is essential for creating technology that is both effective and human-centered. Engineers can design user-friendly products that meet real needs and improve user experiences by understanding how people think, feel, and interact with systems. This knowledge is particularly crucial when developing AI systems and technologies that direct impact decision-making, ethics, and privacy. Behavioral insights help engineers design systems that engage users without manipulation, foster well-being, and mitigate issues like bias in AI. When we talk about AI, bias is a big problem because AI is nothing without the data that we feed. Additionally, as technology plays a growing role in mental health, cybersecurity, and public policy, the interdisciplinary collaboration between engineering, psychology, and other fields ensure that solutions are inclusive, accessible, and most importantly, socially responsible. In today's interconnected world, engineers who are aware of human behavior are better equipped to create innovations that are not only technically sound, but also ethically aligned and responsive to the societal challenges we face. Another point I feel strongly about is the role of women in this evolving landscape, especially in the STEM domains. In my own experience, I have seen how women in tech have the potential to drive innovation that leads to economic growth. According to International Finance Corporation and World Bank 2023 information, the presence of women in Sri Lankan boardrooms is below 10%, which is lower than in India, Bangladesh, and even lower to Pakistan. Even though 52% of the population in Sri Lanka is female, over 91.6% of them are literate, with 32.1% participating in the workforce. And I think we all know who get the most prizes in the universities, right? As Sri Lanka navigates this economic crisis, addressing the gender gap in STEM fields is more crucial than ever. With an increasing number of jobs being digitalized, it is essential that women are not left behind in this transition and contribute to the development of a more equal and sustainable future economy. Women in Sri Lanka's workforce predominantly belong to the low-skilled and informal workforce and continue to be a minority in sectors like the tech industry, for example, which is one of the fastest growing and highest paying sectors worldwide. Closing the gender gap in STEM will ensure that women have access to more high-skilled employment and are meaningfully included in the economic growth that is driven by technology and innovation. At the same time, as these fields evolve and expand, it is increasingly imperative that the capabilities and perspectives of women are brought into bring about innovative, inclusive, and effective solutions to the complex societal challenges of today and to strengthen our collective performance on economic and development indicators. During the COVID-19 pandemic, for example, it was largely women in medical research who led the development of vaccines while also studying the gendered impact of COVID-19 on women's bodies, a nuance which is too often excluded from scientific studies. In a time of growing online violence, women in tech are leading the advocacy and work on developing safeguards and accountability systems to protect female users. As work on artificial intelligence expands, women's technical contribution and oversight is vital to combat gender biases in AI algorithms. We cannot have products and innovations that reach out to all communities, genders, and consumer groups unless women equally drive them. Closing the gender gap in STEM in Sri Lanka will require targeted and continued efforts in society, institutions, and the operational framework of STEM fields. We must deconstruct harmful gender stereotypes and biases on the role of women in society and their capacity to thrive in and expand STEM fields. But what does that mean? It means holistic efforts to recognize our own individual assumptions and biases about each gender and to address them in our daily lives. It means reforming our educational structures and approaches. Our schools and universities must allow and encourage students to explore their interests regardless of their gender and expand from current approaches to, for an example, encourage boys to explore domestic skills and the arts and girls to explore mechanics and high-intensity sports. Our educational structures must celebrate those who have crossed gender lines and pave the path for innovators, scientists, and artists alike. Curricula across all levels of education must build an understanding of social issues and critical thinking skills to recognize and deconstruct biases and discrimination. Academics must increasingly examine issues of science, technology, engineering, and math through the lenses of gender, which aligns with growing research that makes vital discoveries when adopting sex and gender disregarded analysis. Research in gender studies, economics, and technology must continue to inform policies that support inclusivity and diversity, ensuring that women are equally represented in fields that shape our economic future. As I conclude, be bold in your research, be fearless in crossing disciplinary boundaries, and be mindful of the societal impact of your work. The future of economic development is not the responsibility of one field alone, but a collaborative effort across sectors, disciplines, industries, and communities. Our research has the potential to spark innovations that can drive economic growth, shape policies, and create a better future for all. As you move forward in your disciplines, remember that your work does not exist in isolation. Whether you are working in the fields of AI, economic, public policy, or sociology, your contributions are part of a larger tapestry of knowledge that can transform societies. Thank you, and I look forward to seeing the incredible researchers that will emerge from this conference and beyond. Let's continue to push boundaries, innovate, and contribute to a future where economic development is driven by thoughtful, multidisciplinary collaboration. Thank you.
Thank you very much, Ms. Chatani, for the wonderful insights provided. Now I would like to invite Mr. Lakmal to present the token of appreciation to Ms. Chatani. Ladies and gentlemen, we will be starting the sessions within 4 minutes' time. Thank you.
Good morning, ladies and gentlemen. On behalf of Organizing Committee, Faculty of Graduate Studies, University of California, we would like to welcome you to 24th International Postgraduate Research Conference, IPRC 2024. This session has 5 abstracts listed for presentation. 10 minutes are allocated for the author to present their abstract and 5 minutes are allocated for Q&A session. When you have 2 minutes left, we will notify you ringing the bell once and twice to indicate when the 10-minute presentation time has ended, and ringing 3 times to stop presenting. The session chair will be then conduct the Q&A. Your cooperation and conducting the session productively and much appreciated. It's now my pleasure to briefly introduce the session chair, Professor Nimanthi Jayatilaka, Department of Chemistry, University of California. We would like to hand over the session to Professor Nimanthi Jayatilaka. Over to you, madam.
Mic work? Right? Can you guys hear me? All right. We'll just start off right away. The next presenter is BKHH Batuvita. The title of the presentation is Antioxidant Effect of Curry Leaves, Mariah Carnegie, Power on Acrylamide Mitigation in Spring Rolls Covered with Breadcrumbs.
Good morning to you everyone. I'm Hansini Batavita. So let's discuss about the presentation on antioxidant effect of curry leaves powder on acrylamide mitigation in spring rolls covered with breadcrumbs. This is the outline of my presentation. Have you ever heard about acrylamide formation in food? Acrylamide is considered as a food toxicant which is formed during high temperature associated food processing. Especially, International Agency for Research on Cancer has declared acrylamide as a probable human carcinogen. Apart from that, acrylamide can cause neurotoxicity and also other toxic association in our body. In here you can see acrylamide formation pathways. Acrylamide formation occurs through free radical mediated reactions. The precursors between reducing sugars and amino acid asparagine, acrylamide formation occurs. According to European Union benchmarks, several food items had been categorized under this approved acrylamide contents. In here you can see the highest acrylamide contents are recorded in fried potato products and roasted coffee products. However, majority of foods have not yet been categorized under these segments, and it's some sort of a drawback when making decisions. So, my research objectives were to evaluate the ability of curry leaves powder in reducing the acrylamide formation in spring rolls covered with breadcrumbs, and also to evaluate the ability of curry leaves powder incorporation into foods and to improve the fried oil quality. Let's move on to the methodology. The standard recipe was developed for these spring rolls covered with breadcrumbs, and also we simply identified that spring rolls covered with breadcrumbs as fish rolls. So here on notes I will use the word fish rolls in the future. These fish rolls were fried in deep unrefined coconut oil at 170 degrees Celsius for 10 minutes. When using curry leaves powder incorporated samples, curry leaves powder was incorporated into both inner part and the outer part of the sample. The concentrations I have used here was 0.5%, 1% and 2%. In here, these are the parameters I have analyzed for this study. Especially, the acrylamide content in the food was analyzed using liquid chromatography tandem mass spectroscopy, and deuteroacrylamide was used as the internal standard. Acrylamide content in fried oil was analyzed using high-performance liquid chromatography. In here, methacrylamide was used as the internal standard. pH determination was done using pH meter. Total phenolic content, ferric reducing antioxidant activity, and ABTS total antioxidant activity, also the flavonoid content was determined using spectrophotometric methods. Color parameters was done using colorimetry, Loibon colorimeter. The sensory analysis was done using 30 semi-trained panelists. In here, 5-point hedonic scale was done to identify the sensory properties of the foods. Let's move on to the results and discussion. In here, you can see curry leaves powder has significant amount of phenolic content, antioxidant properties, and flavonoid properties. When incorporating curry leaves powder into both inner part and the outer part of the fish rolls, it has significantly improved all 3 parameters, including total phenolic content, antioxidant properties, and also the flavonoid content. And higher the curry leaves powder incorporation into food, higher the antioxidant and phenolic content in our food. Especially 1 and 2% curry leaves powder incorporation had gained the highest overall incorporation, has improved its total phenolic content and antioxidant properties. In here you can see, according to the web diagram for sensory analysis, in the aroma value, color value, texture, overall acceptability has tested in here. According to analysis, 1% curry leaves powder incorporated sample had achieved the highest overall acceptability. Considering both overall acceptability from sensory analysis and also the antioxidant properties, 2 samples had been selected to determine the acrylamide content in our food. So in here you can see 1% and 2% curry leaves powder incorporated samples had significantly reduced the acrylamide content in both fried food and also in the fried oil. Especially when using the 2% curry leaves powder incorporated sample, it has almost reduced the acrylamide content by 50%, and it's almost very important when it comes to acrylamide reduction. In here you can see, when adding curry leaves powder into fish rolls, it has significantly reduced the acid value, free fatty acid content, and also the peroxide value of the fried oil. Since curry leaves powder contains huge amount of antioxidants, it has affected on the peroxidation process, and it has reduced the peroxidization of the oil. This has led to significant reduction in the acid value and peroxide generation, reducing the peroxidation. According to correlation analysis, total phenolic content, antioxidant tests, and flavonoid content had shown strong negative correlation with the acrylamide content in food. And also, acrylamide in food has shown a very strong positive correlation with the acrylamide in food. Since curry leaves powder is having high amount of antioxidant, it will react with the free radicals generated during this acrylamide formation. So in that case, since the free radicals are stabilized with the reaction with antioxidant, it will reduce the formation of acrylamide content. Moreover, this acrylamide, when acrylamide is forming, I have already mentioned that asparagine is important since it's the main precursor. And this antioxidants will react with the asparagine and it will not produce acrylamide, but it will generate other compounds which do not support acrylamide formation. According to moisture content, color values and the pH value, curry leaves powder incorporation had not significantly affected on the moisture content of the food. However, it has reduced pH value to a certain level, and also it has reduced the color parameters, especially in A value and the B value. Since the curry leaves are given some sort of a dark color to the food, this A value and B value had reduced. So, I would like to conclude that this Curry leaves powder incorporation into fish rolls has the potential to reduce the acrylamide content in both fried oil and also a fried food. So when it's added into our food adequately, it can reduce the oil peroxidation and also it can improve the oil quality as well. Since this acrylamide formation can be hindered due to the reactions between antioxidants and the free radicals, the acrylamide formation can be mitigated to a certain level and it can be acceptable to our body. So, I would like to thank the Research Council of University of Sri Jayawardenepura for supporting financially in this research. And these are some references I have taken into consideration for my study. And thank you.
Thank you, Ms. Batuita. The session is now open for questions.
Interesting research. I have one question from you. Is there any particular reason why you add curry leaves powder to the inner mixture? Because of course we can understand why you put it into the outer pot, but what is the purpose you add it to the inner mixture?
Mainly, our Sri Lankans prefer curry taste, especially when they always consider about the curry leaves taste in the middle part as well. Because when only it's eaten from the outside, it will give that taste and the flavor, but it won't give that specific flavor in the middle part. So it's also added into both parts, especially inner part and the back inner part and the external part. And also, it's assumed that since the temperature is almost higher in frying any kind of deep fried food, it will affect the middle part as well. When it comes to acrylamide formation, it will not only limited to the inner part, but also external part, but the inner part as well. There will be some sort of acrylamide formation in the inner part. So that sort of acrylamide content formation can also be reduced when the some amount of powder is inside as well, because the reactions will occur that area as well.
So do you have any record of like how the heat penetrates, like what thickness that a particular heat can penetrate through the layers? I wonder because it's going to be kind of a thick layer of starch material. So maybe, but do you have any evidence in literature regarding this purpose?
Normally, in this study, I have used 170 degrees Celsius for frying. However, when I put some, we have to consider the inner temperature as well. And then I determine it will about 120 degrees Celsius. It is adequate enough to form acrylamide in food.
Thank you.
Thank you.
You didn't see a good correlation between acrylamide formation in oil, heated oil, correct? Acrylamide formation in oil in the presence of curry leaf powder, you did not see a correlation, right?
It has reduced the, it has...
No, I'm asking about the correlation study that you did.
I have determined the acrylamide correlation, I think.
Can you go to the slide? So this oxidation to form acrylamide, that is for the amino acid oxidation, it's from amino acid oxidation?
No. Amino acid is a precursor for the acrylamide formation, and...
Okay, why did you look for acrylamide formation in coconut oil?
Coconut oil.
Did you already measure the amino acid content in coconut oil before you tested the acrylamide formation?
No, I didn't check the amino acid content in the oil. I take it to this study because especially I have done a survey in Colombo district, especially in market survey. The restaurant people always use either palm oil or unrefined coconut oil. Since the price of that unrefined coconut oil is very low considered to that, it was Actually, it differs time to time. When there's some sort of importing barrier, there will be weight difference and cost differences. And that time, the unrefined coconut oil was the mostly used frying oil by the market people. So that's why this oil was chosen for this study.
I think your study is good, but there are some gaps that you need to start filling. One is you're saying acrylamide formation happens because of the amino acids present in the food. Then you need to check for the amino acid content in the oil that you choose. Second thing, refined coconut oil is cheaper. It's not more expensive, and coconut oil is a saturated oil. It is pretty stable. You're looking at oxidation of oil, so it's better to look at other oils. Third thing is, coconut oil is rich in antioxidants. So if you're trying to look for correlation of effect of added antioxidants on that oil, you need to strip the oil first, either that or choose an oil that is commonly used. Okay?
Okay.
Thank you.
Thank you.
Okay, the next presenter is Miss. Is it H.M. Dilruk or is there another name written here? Okay. Our next presenter is H.M. Dilruk. The title of the presentation is Evaluation of the Fitness of RNA Interference-Based Aedes aegypti, Linnaeus, Transgenic Mosquitoes.
So this is our findings on evaluation of the fitness of RNA interference-based Aedes aegypti Linnaeus transgenic mosquitoes under the main project of high-tech transgenic approaches to control dengue vector Aedes aegypti by genetic engineering. So this is the outline that I am going to present in today. So as the introduction, as you all have known, the dengue is a rapidly emerging viral disease caused by the 4 distinct serotypes of dengue viruses, and approximately 390 million infections occur each year worldwide. Due to the unavailability of the effective vaccine or the proper treatments, disease control primarily relies on the vector control strategies. But among them, those conventional vector control strategies have not been proven effective 100%. So as an innovative approach, transgenic mosquitoes have become prominent. So among them, RNA interference techniques provide a versatile means of modulation in the transgenic mosquitoes. So this is RNAi is an evolutionary conserved gene silencing pathway that presents in the innate immune system of the mosquitoes. So there are also, as you can see, the short hairpin RNA will be cleaved by the RNase 3 enzyme. Then the small interfering RNA will be incorporated into the RNA-inducing silencing complex. Then also, in order to target the mRNA molecule of the virus RNA. So there are a polysystemic cluster of 9 synthetic small RNAs was designed in order to target all the 4 dengue viral serotypes in a previous study. So in this schematic illustration, you can see the design transgene construct. So there, the dengue viral target sequences also can be present. So when considering about the transgenic mosquitoes, their fitness has to be measured. So when we are talking about the fitness, their developmental fitness as well as the reproductive fitness has to be taken into account. So when it considering about the development fitness, their longevity, wing length, as well as the reproductive fitness, we have to talk about the fecundity as well as the male mating success as well as egg hatchability. So there, the transgenic mosquito should have sufficient fitness in comparison to the wild-type counterpart to be used as effective vector control method in population replacement strategies. So there in our research project, what we have done is that the fitness evaluation of this newly developed RNA interference-based Aedes aegypti transgenic mosquitoes were evaluated under the laboratory conditions. So here's the research objective of our research project. So we have evaluated the fitness of this newly developed RNAi-based transgenic mosquitoes. They are the fecundity, hatchability, male mating success, standard wing length, as well as the flight ability and the finally longevity were assessed. So in the methodology, a hemizygous line of the mosquito transgenic line was obtained, and the morphological identification was carried out using a fluorescent microscope, and the red fluorescent protein expression was observed in the fourth installer layer of the mosquito eyes. So there, the mosquitoes were maintained at the laboratory containment, arthropod containment level 2 facility at the Molecular Medicine Unit, Faculty of Medicine, University of California. So both transgenic mosquitoes as well as the wild-type mosquitoes were fed with the 10% glucose solution, while the females were fed with the cattle butt to obtain the eggs. And both of these fitness parameters also were assessed in the same laboratory conditions. And when we are measuring the fecundity, the number of eggs laid by each and every mosquitoes were divided by the number of mosquitoes. So in the methodology, hatchability was calculated as the percentage of larvae hatched from the total number of eggs laid, and the male mating success also were assessed by mating one male transgenic or wild-type mosquito by mating with the 5 female wild-type mosquitoes. So there are also the 20 replicates were used, and so you can see in this image the standard wing length of the transgenic mosquitoes as well as the wild-type mosquitoes were assessed from the distal end of the alveolar to the end of the radius wing. So both females and the males wing length was measured, and the flight ability measuring equipment that has been kept in the inside of the mosquito cages can be seen in there. So there are also 30 pupae was used to assess the flight ability, and we have counted the number of emerged adult mosquitoes that came out from this 25 centimeter transparent tube from 96 hours. And the longevity of male and female transgenic mosquitoes as well as the wild-type mosquitoes also was evaluated using 90 mosquitoes as the total, and also the number of mosquitoes were counted daily in each cages as 3 replicates separately until the death of the last mosquito. So this is as the results and the discussion. So here in this A arrowhead shows you the fluorescent eye expression of the larvae of the transgenic mosquitoes, whereas the B shows you the wild-type mosquitoes eye color expression. So in the fecundity and hatchability, there were no such significant difference in between the transgenic as well as the wild-type mosquitoes in comparison. So the 2 sample t-tests was used to analyze the fecundity difference between the transgenic and the wild-type mosquitoes, and as well as the male mating success also were assessed with reference to the percent of egg hatch, and there also the significant difference was not observed between the transgenic and the wild-type mosquitoes. So the standard wing length also, a significant difference was not observed even in the transgenic and the wild-type mosquitoes, even when comparing to the females as well as in the males. So finally, and the flight height ability measurement, but there was a significant difference of the flight ability of the female transgenic mosquitoes when compared to the wild-type mosquitoes, and this research indicated that the flight ability of the female transgenic mosquitoes was significantly reduced compared to the female wild-type mosquitoes, and also, however, flight ability of the transgenic mosquitoes was not significantly different from the wild-type mosquitoes. So in the research and discussion, so here you can see the mean values of the transgenic mosquitoes flight ability and the average number of mosquitoes with 25 centimeter of the flight ability. So then the evaluation of the longevity. So in the longevity evaluation also, a significant difference was not observed between the average longevity of the male transgenic mosquitoes and the wild type mosquitoes. And when it comes to the females as well, between the transgenic mosquitoes and the wild type mosquitoes longevity, a significant difference was not observed. So here you can see the results of the longevity assessments. And finally, in the conclusion, this study concludes that there was no significant effect because of the most of the fitness parameters, such as the fecundity, hatchability, male mating success, standard wing length, and the longevity. But however, a negative effect on the flight ability was observed. And the absence of such a significant difference in most of the fitness parameters that we assess confirm that the RNA interference-based this transgene construct has a minor impact on their mosquitoes' fitness. So we would like to acknowledge to the accelerating higher education expansion and the development operation, as well as to the International Atomic Energy Agency for the technical cooperation. And we would like to acknowledge to the Molecular Medicine Unit to facilitating this carry out this research project. So finally, these are the references that I used to create this presentation. And finally, thank you for your listening.
Thank you. This session is now open for questions. It's possible that I may have blanked out when you explain this, but can you explain, first of all, what this RNAi target, and secondly, why we care about the fecundity and longevity of these transgenic breeds?
Yeah, of course. So this RNAi targets the dengue viral of all the 4 dengue viral serotypes. So when we are designing a transgene construct, this RNAi has to be designed in a way as effective for all the 4 dengue viral serotypes. So this RNAi mechanism, the transgene construct that has been inserted into the mosquito genome can produce siRNA with the 9 siRNA, obviously. So it has the capability of binding with whatever the mRNA that comes to the the mRNA of the dengue virus. So in all the 4 serotypes can be targeted by using this transgene construct. And as for your second question, so we do this fitness assessment just because of releasing these transgenic mosquitoes into the wild, and we need to know whether there are some fitness reduction happens due to their transgenesis.
Why do we care?
Of course, yes, because if we create the transgenic mosquitoes with the low fitness, and if due to the transgenesis, if their fitness has been reduced, there is no point of producing such a transgenic mosquito. So these transgenic mosquitoes also should have the same capability with the wild type mosquitoes in order to compete with them for the reproduction as well as for the mating with them. Then only they can propagate their population for the next generation. This is kind of a population replacement strategy. In order to replace the population, they should have the sufficient fitness compete to their wild type counterparts. Otherwise, they won't be success in the field.
All right, the last question. The RNAi that you have designed, yes, is it targeting the viral mRNA or is it targeting the viral genome?
It is targeting the viral mRNA. So
which ones?
Viral mRNA.
You don't know the target?
Exact target but still we haven't published it, the sequence. So this sequence has been designed in a way that is complement by choosing the complementary or the RNAi genes of the mRNA of the dengue virus. So this work, so still these sequences haven't been published yet, so I'm not supposed to tell about the sequence details.
Okay, all right, thank you. Any other questions? Thank you. The next presenter is APH Somaratna. The title of the presentation is Fungal and Plant DNA Extraction Using Cellulose-Based Paper Strip Method.
Good morning, all of you. I would like to present my research on fungal and plant DNA extraction using cellulose-based paper strip method. Let's move to the introduction part. The polymerase chain reaction, or the PCR, is a frequently used laboratory technique, but the extraction of pure DNA from fungi and plants for PCR and sequencing purposes is very difficult due to compact cell wall on these species. When we talk about Aralia leaves contain latex, and orchid leaves are fleshy plant leaves. And also, these plant leaves contain high polyphenol content, polysaccharides and secondary metabolites. So those secondary metabolites interfere with DNA and inhibit the PCR reaction. So this research was conducted to check whether the impurities can be removed from the recovered DNA using the cellulose-based paper strip method and to obtain clean DNA for PCR and sequencing. Let's move to the methodology part. Mainly there were two DNA extraction methods, classical CTAB method and paper strip method. This flowchart shows the main step of classical CTAB method. To check whether we can obtain pure DNA, we have incorporated paper strip method into the several step of classical CTAB method. As you can see in this flowchart, paper strip method after cell lysis considered as method 2. Paper strip method after first organic extraction considered as method 3. Paper strip method after organic extraction after organic extraction following chloroform isoamyl alcohol after adding chloroform phenol chloroform isoamyl alcohol step considered as method 4. Let's move to the results and discussion part. These are the PCR curves that I have obtained. This figure shows CTAB method amplification plot and melting curve for CTAB method for Candida albicans. This figure shows amplification plot and melting curve for orchid leaves by CTAB method. This figure shows paper strip method after organic extraction for Aspergillus niger. This figure shows amplification plot and melting curve for paper strip method after organic extraction for Candida albicans. This figure shows amplification plot and melting curve for Candida with paper strip method after double organic extraction. This figure shows melting curve and amplification plot for Aralia with paper strip method after double organic extraction. As you can see, CQ values of curves given by these two methods are less than 30. That means CQ values less than 30 are strong and indicative that there is abundant target nucleic acid in the sample. These CQ values tells how many cycles it took to detect a real signal from the sample. Melting temperature is the temperature at 50% of the DNA double strand separates. In this study, melting temperatures of these curves have melting temperatures in between 81 to 89 Celsius. In this study, better PCR curves were given for Candida, Aspergillus and Niger, and Aralia leaves with paper strip method. For only orchid with CTAB method, better PCR curve was given. Though these methods have low purity ranging from 1.1 to 1.5. When I talk about fungal species, better PCR curves were given for both fungi with paper strip method, but a better PCR curve was given for Candida only with the CTAB method. That may be due to 2 layers of melanin on the cell wall of Aspergillus niger, or the black color pigment of Aspergillus conidia. The black color pigment co-extracted with DNA, and that may be interfere with DNA and inhibit the PCR reaction. But when we talk about paper strip method, the black color pigment and other secondary metabolites were removed by paper strip method and gave a better PCR curve. That means a single washing step is enough to remove those impurities. When I talk about plant species, better PCR curves were given for orchid with CTAB method, but not for Aralia. That means maybe due to high oxidized polyphenol content found in Aralia leaves. That was noticed when I doing the extraction. A brown color DNA pellet was recovered. Therefore, those impurities did not remove with the CTAB method, but in paper strip method, with the double organic extraction and the single washing step, useful to enough remove the impurities in paper strip method and gave a better amplification curve. In conclusion, the purity of DNA from all 4 methods for each species did not show any significant differences. So the paper strip method can be used as a substitute method for CTAB method and to obtain clean DNA for fungus and plant samples. Thank you.
Thank you. The session is now open for questions.
So you have used a paper strip method. So
Yes, madam.
what do you mean by paper strip method? How do you do that?
Paper strip method is the paper waterproof handle and nucleic acid binding area combined. It prepared by Whatman filter papers. The waterproof handle made by paraplast layer, covered with the paraplast layer.
So it's that we can prepare that paper strip in our laboratories?
Yes, madam.
Okay, so without any other high-tech invention is not necessary.
Yes, madam, not necessary.
And have you tried other specific plant species, like maybe like waxy kind of plant species to
No,
see?
no, madam, I have only checked Aralia and orchid leaves.
And what was the purity you obtained?
The purity obtained was 1.1 to 1.5. The CTAB method purity was 1.5. That means there is no any difference between those 4 methods.
Okay, thank you.
Since you failed to explain why this is interesting, and now that you have explained, you make the paper strip by using Whatman filter and overlaying paraplast to make the handle. Can you explain how long it takes to do a DNA extraction using this paper strip method?
This paper strip dip 1 minute into the cell lysate and rotate it to bind DNA, and then tris bases the washing buffer, and then the paper dip into the washing buffer, and elute DNA into nucleus-free water.
So how long? How long does it take for you to do that? You lyse the cell sample, and then how long does it take for you to do a DNA extraction?
This method is developed for field activities. Actually, I have tested this method whether we can obtain pure DNA.
Familiar with the term yanne koyeda malle pol? How long does it take for you to do a DNA extraction? Is it 1 minute,
Using
2
paper
minutes?
strip method?
Yes.
It takes nearly 3 minutes.
All right, thank you.
Thank you.
The next presenter is Eche Niluni Ruvishani Samendi. The title of the presentation is Optimization of RNA Extraction Method from Latex of the Rubber Tree, Hevia brasiliensis, for gene expression analysis by RT-PCR.
Thank you. Good afternoon, everyone. I am Niloni Rushani Samindi, and I will be here presenting on optimization of RNA extraction method for from latex of the rubber tree, Hevia brasiliensis, for gene expression analysis by real-time PCR. Let's carry on the presentation using these outlines. Rubber tree, scientifically we call it as Hevia brasiliensis. It is a key species in the global rubber industry, providing natural rubber, which has many economical applications. Despite its economical importance, especially in Sri Lanka, there are very limited researchers have done in molecular studies. So there is a vacuum in this area. Typically, we use for RNA extraction immature leaves of the rubber tree. But when in the lysis step, when we crush those immature leaves, the polyphenols and other metabolics like proteins, lipids, and polysaccharides are mixed with RNA, and they co-precipitate with
the RNA in the final step. So, those inhibitors, those contaminants inhibit PCR reactions, and it's cause for RNA degradations, and also challenge for the reliable results. If we briefly go through the RNA, ribonucleic acid, a single stranded molecule involved in various cellular functions and act as a messenger. There are three main types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. And also some researchers found that other RNA types like miRNA and siRNA, but they are in very low amounts in the RNA isolation is more important in gene expression studies, cDNA, library constructions, cloning, RNA sequences like this. As I said before, RNA's stability issues and sensitivity to contamination may inhibit and impact for experimental accuracy. Therefore, my research objective is to develop and modify a precise, efficient, and reliable method for extracting pure, high-quality RNA from the latex of the rubber tree that is suitable for downstream application, ensuring accurate and reliable gene expression profiling. Our methodology is done by mainly three steps. First, I have collected latex samples from the department nursery at the 10 years old plants. As precautions, we used sterilized working area, sterilized equipments, and also cold chemicals and cold conditions for transportation. There are three steps mainly. We extracted RNA from both existing method and optimized method, and then monitored and validated using RT-PCR. Here you can see my optimized protocol. We introduced critical modifications to this as additional chloroform extraction step, multiple ethanol washes, and also freeze-drying RNA pellet at -20 centigrades overnight. Then it is monitored by using 1.8 agarose gel electrophoresis and nanospectrophotometric method. After that, those RNA was validated by using gene expression method. There we use RefGene for that is responsible for latex production in the rubber tree. There we use cDNA, which are used by both existing optimized methods, isolation RNA. Let's see, move into the discussion part. Here you can see here we compared both existing method RNA and also optimized method RNA by visualizing agarose gel visualization. In the first image, you can see our existing method extracted RNA, there you can see the bands are not clear and they have more contamination and degradations, and the bands are smeared. In this second picture, our optimized method, there the bands are clear and no contaminations, no smears. The bands are clearer than the existing method. Then we monitored them through the nanospectrophotometer. Here you can see the red colored highlighted ones are the optimized method one. Their concentration of the optimized method RNA is higher than the existing method RNA concentration, and also those almost all samples showed A260 to A280 ratio around 2 and A260 to A230 ratios between 2 and 2.2. So those values indicate excellent purity free from proteins and phenol contamination and higher contaminations. Further, we validated those RNA from gene expression analysis. In the first two images, we used, you can see the real-time PCR we used in using existing method RNA, and the second one's are optimized method. In the second picture, you can see very consistent melting curve and...
Time's up. Can you wrap up?
Sorry?
Can you wrap up? Wrap up doesn't mean you're going to keep talking. Just say whatever, the final slide, and then move on. We can see it. Just conclude.
Okay. As the conclusion, additional, our optimized steps, additional chloroform extraction method, multiple RNA pellet washers, and freeze-dried RNA pellet help to mitigate RNA degradation due to the RNA mitigations. Overall...
Okay, thank you. The session is open for questions.
Good afternoon. This research, what is the significance of this research? Because nowadays, faster RNA extraction kits are available in the market. Yeah. So these are the, you are modifying some other old traditional method. There's so many steps involved in there. In the kits, you will buy a kit, you will have 4 or 5 steps. You can isolate far better RNA on this. I am working on, I already work on more I usually work on kits. So what is the significance of this research? If you are telling that waxy, thick plants, latex are impurity, but the kits you will have specific for that plant. So what is the significance of the research? So you will take a whole long day to extract this RNA. In kits, you will get half an hour, you will finish the research.
Yeah, first of all, we have actually financial problems in the department to use so many kits. There are so many students in the came to the department for researches and trainings and so on. So actually they couldn't buy those RNA kits.
Yes, okay, I understand. In Sri Lanka, we have like lack of grants. Okay, so no problem.
Any other questions? You keep saying, you showed the gel and said that it reduced contaminations. How did you draw that conclusion?
Sorry?
You showed the gel, and then you said in the gel, you can see that the new method reduced contaminations. How did you draw that conclusion? What is that conclusion based on?
Because the gel bands there is no smears and the degradations at the bottom of the gel. They are in very low amounts.
So what are the contaminations that you're referring to?
There may be protein contaminations and phenol contaminations.
I think you're a little confused about what can be seen in a gel. I understand that you're saying there's less degradation, and maybe you might be saying that it is because RNA's component might be removed during your extraction procedure.
Yeah.
But the conclusions you drew regarding phenol content, you cannot draw any of it based on the evidence you have shown here. There is not a single step during your process where you have shown the phenol content used in the old method, the presence of phenol content, and you have not shown that the phenol content is not there anymore after using the new method. So you can't draw conclusions that you don't have evidence for. You are not showing us any evidence of removing phenolic contaminants. In order for you to say that, you have to show it. Okay? All right, thank you.
Thank you.
The next presenter is Tennakon Mudyanselage Oshadi Dilshani Tennakon, optimizing the efficiency of a probe-based duplex qPCR assay for accurate detection of normalized PI4K Ka gene copy number.
Good afternoon, everyone. My name is Oshadi Tennakon, and I'll be presenting my abstract on optimizing the efficiency of a probe-based duplex qPCR assay for the accurate detection of the normalized PI4K gene copy number, which was done under the main research of developing a diagnostic test to detect a microdeletion that is commonly found in children with neuropsychiatric phenotypes. So, starting off. This will be the outline of my presentation. And to talk about quantitative PCR, which is one of the most commonly used molecular diagnostic tools in Sri Lanka, in order to detect gene expression, measure gene expression, detect genetic mutations, and all sorts of activities. And it is a very powerful tool. And the ability of quantitative qPCR to be multiplexed makes it even more powerful. But here's where the challenge comes in. Despite the ability to be multiplexed, once the qPCR reaction becomes multiplexed, this impairs its PCR efficiency and reduces the balance between the amplification efficiencies. In my research, since we were measuring the DNA concentration of a target gene relative to the DNA concentration of a reference gene, maintaining balanced amplifications between each of these genes was critical. So, keeping that in mind, my research objective was to optimize and validate this duplex qPCR assay, which uses TaqMan probes to simultaneously detect the gene concentrations of the PI4Ka gene, which was my target gene, and SHANK3 gene, which was my control gene actually, aiming to achieve balanced amplification between these two genes and to reliably quantify these two genes. The monoplex reactions for these two, for the detection of these two genes was already optimized, and the they were optimized for the DNA concentration, magnesium concentration, and for the individual primer concentrations. Using these conditions, initially, two monoplex reactions and two monoplex reactions and a single duplex assay was done using a DNA dilution series, a 10-fold DNA dilution series ranging from 100 nanograms to 0.1 nanogram. And using the data obtained in these reactions, 3 standard curves were generated, and the reaction efficiency was calculated using the formula shown in the screen. All the reactions were done in duplicates using the CFX machine in order to ensure reproducibility. So these two curves were the ones we obtained for the monoplex reactions. Generally, the acceptable range for PCR efficiency is between 80% to 110%. So both the monoplex reactions showed PCR efficiencies that were within this acceptable range and had balanced amplification and we could use these standard curves to reliably quantify these two genes. But moving on to the duplex reaction, what we observed was the two genes were run in two separate channels, FAM and HEX. What we observed was the HEX channel had a amplification efficiency which was out of the range 80 to 110%, and the correlation coefficient was also extremely low. So this showed that the amplification was imbalanced, and this could have been mainly due to the competitive nature between the two primers, where one of the primers overpowered the other during the amplification process. So our next step was to optimize the primer concentrations for the duplex PCR. And different primer concentrations for the PI4K gene, which was the overpowering gene, overpoweringly expressed target gene, and the control gene SHANK3, which had lesser expression, was used. Different concentrations were used, where low concentrations were used for the overpowered gene and high concentrations were used for the control gene. And finally, the lowest CT values having the highest amplification was selected as the most ideal primer concentrations for the duplex PCR. And this concentration was used to generate another standard curve where their amplification efficiencies were calculated. And then finally, the accuracy of the assay was tested by using control and positive samples and calculating their gene dosage values. So these were the standard curves that we obtained once the primer concentrations were optimized, where we obtained a good efficiency, amplification efficiency for both the channels within the range of 80 to 110%, and the correlation coefficients were also of commendable values. Using this optimization, the primer optimization, we were able to optimize the duplex assay to obtain balanced amplifications and reliable quantifications. And that brings me to my conclusion. So using qPCR, we can do a lot of gene detection, measuring gene expression, and but when it comes to multiplexing, there might be drawbacks where the amplification might not be balanced. So optimization is further required in order to bring about balanced amplification and reliable quantification. So in my reaction, SHANK3, the control gene, required a primer optimization where we increase the primer concentration to 0.4, 0.8 micromolar concentration, and PI4K, the target gene, was decreased to 0.2, 0.2 micromolar primer concentrations in order to produce the robust and accurate assay to detect this target gene. But further validation against the standard method is required for this test to be used in clinical situations. And I would like to acknowledge these people for aiding me in my research. Thank you, and these are a few references I used.
Thank you. The session is now open for questions. What was the reason you gave for this change in the amplification efficiency?
If in the initial stages, madam?
Yes.
When a PCR reaction is done as a monoplex, generally they have high amplification efficiencies. But when it's duplexed, due to the competition between the primer pairs, when there's multiple genes to be amplified, the efficiency tends to reduce in probably the one, or if there's multiple, there might be several genes that are having less amplification efficiencies.
Did you look at the primer-dimer formation? Have you done a melting curve analysis?
No, madam, I didn't do a melting curve analysis.
You check the literature. One of the reasons for high amplification efficiency than expected is primer-dimer formation. Since you're doing qPCR. Yeah, but you're doing qPCR.
Yeah.
DEL does not have the sensitivity then the qPCR melting curve analysis would have. So I think it's a good idea to take a look at your melting curves.
Yes, thank you so much.
Any other questions? All right, thank you.
Thank you.
If there's no more questions, I think we're done with this session. Am I right? Yes?
Thank you very much, all the presenters. Now I would like to invite the session chair, Professor Nimantha Jayatilleka, to award the certificates for the presenters. So please. Right. Thank you very much. So I'll invite Professor Deepika Amarasinghe and Professor Suranga Vikramarajchi to deliver the certificates to our presenters. Presenters, please come to the stage as I read your name. BKHH Batuita. HM Dilruk. There are 3 other presenters, and I invite Professor Suranga Vikramarajchi to present the certificates to the rest of the presenters. APH Somaratna. H.A. Niluni Ruvishani Samindi. Oshadi Dilshani Tennuko. Thank you very much, madam. Now, we would like to convey our thanks to the presenting authors for their submissions and the many interesting concepts, ideas, and applications that were set out in different areas. We trust that based on your findings presented here and the discussions, you will be able to improve and further refine your research study. Now, on behalf of the organizing team, we would like to thank our session chair, Professor Nimanthi Jayatilaka, for providing your professional input to the authors and the audience here. And I hope that the discussion will help the authors to refine their work further. Thank you, Professor Nimanthi Jayatilaka, for consenting to be here today. We appreciate your presence, and as a token of appreciation for the hard work and dedication, we have arranged a small gift for you. And I invite our session coordinator, Mr. Pasan Dharmavardhana, to hand over the token of appreciation to Professor Nimanthi. Thank you very much, madam. Ladies and gentlemen, thank you very much. And that concludes the first session of IPRC 2024. Thank you for participating, and we invite you to the next session, which will start at 1:15 p.m. here in this same auditorium. Thank you very much. Good afternoon, ladies and gentlemen. We are about to start the second plenary session of today's IPRC conference. Today, as our resource person, we have an energetic personality, Dr. Patrick McNamara. He will be presenting under the topic of transdisciplinary research, creativity, and innovation for economic development. And to formally introduce Mr. Patrick, I would like to invite Dr. Thelma to introduce. Thank you very much.
Thank you. Good afternoon, ladies and gentlemen. It's my pleasure to introduce Dr. Patrick McNamara, the Executive Director of the United States Sri Lanka Fulbright Commission and a distinguished professional with over 35 years of experience collaborating with universities, governments, corporations, non-profits, and foundations. Prior to his current role, Dr. McNamara served at the University of Nebraska at Omaha, where he taught courses in conflict resolution, sustainable development, and social entrepreneurship, and held the position of Senior International Officer. He is also a governing board member of Initiatives of Change, a global interfaith organization dedicated to building trust across divisions. A recognized thought leader, Dr. McNamara has delivered a TED Talk titled Water Conflict, Water Peace. Academically, he holds a PhD in public administration from UNO, where his research funded by the US Department of Housing and Urban Development, focused on public-private partnerships addressing homelessness. He also earned a master's in conflict analysis and resolution from George Mason University, specializing in international and organizational dispute resolution, and a bachelor's degree in religion from Swarthmore College with a focus on comparative religions and ethics. Therefore, please join me in extending a warm welcome to Dr. Patrick McNamara to give the plenary speech on transdisciplinary research, creativity, and innovation for economic development for Iprc 2024. Thank you.
Thank you for that kind introduction, Professor Thelma. And it's an honor to be here, University of Kelaniya. Thank you, sir, the distinguished chairman of the UGC as well, and of course, a distinguished professor here. Great to meet so many young and energetic professors and students, and I look forward to hearing more from you as we engage together in this next session. This topic of transdisciplinary research, creativity, and innovation for economic development is a timely topic. Here in Sri Lanka, there are many opportunities that present themselves on this beautiful island. The complex problems will be solved with cross-cutting answers. No longer are the traditional narrow disciplines of academe sufficient. What is needed is a culture of creativity and innovation. With the students at this conference inspiring fresh approaches to today's complexities and new policy frameworks to frame tomorrow's challenges, Sri Lanka is positioned to play a major role in the near and further future. Let me talk just a little bit about the Fulbright Commission, about my own engagement here in Sri Lanka, and then hopefully have some discussion with you signed between the government of Sri Lanka and the government of the United States. It is binational and autonomous. Although most of our funding comes from the US taxpayers, we also have some support from the Sri Lankan side, and we hope to raise more money on the Sri Lankan side in the future. The governing board has 3 members appointed by the President of Sri Lanka and 3 members appointed by the US ambassador to Sri Lanka, and I report to that 6-member board of directors, and all of the staff reports to me. I am the first non-Sri Lankan to direct the Fulbright Commission in 72 years. So it is a great honor and a privilege for me to be in this position. And we have to keep both interests, both the Sri Lankan interests and the US interests, at the heart of what we do with Fulbright. Now, as you know, because you are a Fulbrighter, there are Fulbright scholars, Fulbright students, and we have other categories, teachers, professionals, and others who are going to the US from Sri Lanka to learn and teach and do research in the US and then come back to Sri Lanka. That's a very important point. I always say this is not a one-way ticket. You are required, like literally because of the visa, you are required to come back here to Sri Lanka to give back and build up this great country. Likewise, Americans are coming to Sri Lanka to learn from and collaborate with researchers and artists and professionals here in Sri Lanka. We actually have 7, now actually 8, who are coming this year to Sri Lanka, and next week we have the orientation for them coming in country. And they span from a professor of nursing who is at the University of Colombo, Faculty of Nursing, to a musician who's doing documentary film on Sri Lankan musicians. He's at an NGO called Music Matters. Another who's at the International Water Management Institute doing water research. Another, actually the very first time in Sri Lanka that we have a Fulbright National Geographic Fellow. This is a very unique, there's only a couple of them around the world, and it's the first time in Sri Lanka that the Fulbright National, 1 elephant and 1 human, and telling the story of how they conflict and cooperate. And then he will use all of the National Geographic platforms, the magazine, the website, any social media platforms, to tell the story of that 1 human and that 1 elephant. I'm really excited about that project. So that's a little bit about Fulbright. Now, a little bit about me. I first came to Sri Lanka in 1991, when you were just 3 years old. And I fell in love with this country. I fell in love with the people, the diversity, the incredible ecosystem, the complexities. Some of you might remember 1991 was a difficult time in Sri Lankan history. And I also lived into the conflict here. But I have seen so much positive change happen. I've been coming back again and again over the years, and I'll tell you, right now feels like a very exciting time to be in Sri Lanka. Of course, you just had the elections. I'm not going to get into politics here. I am apolitical in my position, and actually in my life, I am a registered independent. So I am not partisan in that sense. But I'll tell you, to see the NPP and the way in which the first ministers and the parliamentarians who are educated people, in fact, many of them educated right here at Kelaniya, right, including the president himself, right? That speaks to a new era. And I think education is going to be very much in the forefront for how to build the economic development of this country going forward. Intellectual property, patents, the way that entrepreneurs are going to be growing jobs. And it's one thing that I hear as I travel this beautiful country. I hear so many of the young people say, I want to leave Sri Lanka. I want to go to Australia or Germany or China or UK or wherever. And it breaks my heart every time I hear it. I say, stay here in Sri Lanka, build this country up. but it's often the economic realities, jobs that are driving them out. It's not that they don't love their motherland, it's that they see economic opportunity elsewhere. But if economic development is really on the rise here in Sri Lanka, we're going to see not only less brain drain, but in fact, more brain gain coming back here. And that's actually one thing that I am particularly interested in, is the incredibly successful Sri Lankan diaspora. Now, I'm not just flattering you in the audience here, but I'll tell you, Sri Lankans in the US, in Canada, in the UK, are some of the most educated, successful professionals who are doing amazing things. I think that the more that we can engage them back here in Sri Lanka, in whatever way, through philanthropy, through job creation, through partnerships, maybe even through moving back, as some brilliant young professors have done, I think it is the future of Sri Lanka. So that's a little bit about this innovation, and let me say a couple of other things from my prepared remarks here. When I was in Omaha, I spent 6 years as the Director of Philanthropic Services at the Omaha Community Foundation. And my job was to work with social entrepreneurs and advise rich families and corporations about where to give their philanthropy, funding social entrepreneurs. One thing that I took away from that is that social entrepreneurship, or entrepreneurship in general, in a community is more about not just individuals, smart individuals, but about creating the ecosystem within the community. It's about innovation, partly happening in universities, but also the tech transfer that turns those brilliant ideas into products in the market, and ultimately corporations that are selling those. It's about risk-taking, and it's about often allowing for failure. I'm going to go out on a little bit of a limb here and say that in Sri Lanka, I don't always see the entrepreneurship mentality, the risk-taking mentality. They say in Silicon Valley, if you haven't failed twice, then you're not a real entrepreneur. You know, the idea is you take a risk and you fail. But often because of societal pressures, because of family pressures, because of economic reality, and I'm not trying to speak here from a privileged position, but I'm saying sometimes those failures are where you actually learn the best lessons. And next time around, you can make it even better. So allowing for more risk-taking and more failures that ultimately lead to successes is one thing that I hope that Sri Lanka is, growing into in this entrepreneurial moment. A final comment about this interdisciplinary conference. I'm really convinced that the complex contemporary issues facing our world today, what we in the public administration and international relations world call wicked problems, cannot be solved by any one person, by any one field, or by any one perspective. My past research looked at the problem of homelessness in America. The United States Department of Housing and Urban Development funded my research, and I was looking at cross-sector solutions, bringing business, government, and nonprofits together to solve homelessness. We need more of that collaborative, interdisciplinary thinking. An artist brainstorming with a scientist to find solutions to poverty. A medical doctor and a dancer researching how movement can rewire the brain. An engineer and a filmmaker collaborating on interactive AI interfaces. This interdisciplinary approach is the future. And to go back to what I do in my professional life, Fulbright is one of the places that nurtures this creative partnership building. If you would like to learn more, reach out to me. My email address is Patrick. Thanks to the organizers of this International Postgraduate Research Conference 2024 for inviting me to participate and learn along with all of you. Hoping that our collaboration makes this world a more peaceful place. Thank you very much. If we have a couple of minutes, I'm going to open up to questions. Is that okay? Do we have a couple of minutes, or are we done? Yeah? Any questions? I want to especially ask students to speak up. Anybody have a question? Yeah? Come on.
Hello, sir. I'm Sashila, second year undergraduate. So you were speaking about developing Sri Lanka and all the research sectors so that we could come from how we are right now to a better position. So I just like to know whether if we are starting something, we should have a plan as well, right? So anything from your perspective that we should change to make it a like researchly evolve the country, something from your, because
what's your major?
At the moment, biological science. So from third year onwards, we'll be specializing on either biochem, chemistry, or...
Got it. So in the spirit of social entrepreneurship, I want to ask you, and I'm looking at the students here, what do you think are the top, I don't know, 3 or 5 social problems, or let me just say societal problems in general, that Sri Lanka is facing today? What are the top 5 societal problems that Sri Lanka is facing today? Yeah?
Yeah, okay. I think it's the mentality that thinking that we don't have the facilities in Sri Lanka, and then we are moving abroad because we have the knowledge as individuals, but still because of economical issues and all, we tend to go abroad and we don't try to start from here.
Thank you. What do you think is the top problem or societal issue today?
I think people have attitude. It's the one thing need to be changed. Yes, answer. People attitude must be changed in Sri Lanka.
Okay.
They need to do. They need to try to do anything.
Yeah.
Right? I think attitude must be changed. In people.
Got it. Thank you. What do you think is the top societal problem?
So I think about poverty or something. So we have to change that. And this, my friend, he also mentioned that the mindset should have changed.
Right.
So mindset should have changed, and poverty.
Very good. Good ones. I'm going to ask some of the young women over here. What do you think are some of the top issues in society today here in Sri Lanka?
Environmental problems.
What?
Environmental,
yeah. Good. No, I'd rather hold it. Thank you. Go ahead.
People's mind.
Yeah. Okay. So the mindset. Okay. Very good. All right. So we've heard some common themes there, and I think that it's very hard to change mindset, right? That's like a generational change. But there are a lot of psychological studies about how society changes and how mindset changes. And just one thing that I'll mention in that, there are transitional moments in history that turn people in mindset ways to look optimistically at the future or pessimistically at the future. And there's actually research done by actually an Omaha, Nebraska-based company called Gallup, which does this world survey on a regular basis that looks at, are people more hopeful or are people less hopeful in different societies? It's very interesting if you want to track this. But it is often moments of change, either political change, social change, environmental change, that are these nexus points when minds begin to think more optimistically. I think, and it's going to take a little lag time to prove this, but I think right now in Sri Lanka is one of those transitional moments. Now, we never know. I mean, God forbid we have another, Easter bombing, or, COVID comes down on us or something like that, right? Anything could happen, right? But I feel like talking, especially to the younger generation, there is really a beginning of a mindset change. You invite me to the back here again next year, and we'll talk more about that. But I think that right now is a hopeful moment, and that mindset may change. To your point, you said environmental issues, and this is one that unfortunately Sri Lanka isn't going to be able to solve alone. This is a global issue, right? And looking at the United States and some of the other really big polluters, and we saw at COP29 that just happened a couple weeks ago, yes, a little bit of action and a little bit of money pledged from some of the rich countries to give to some of the poor countries, but that is just a drop in the bucket for what is actually needed to change the environmental issues. I think that the issue of poverty is one that I have thought a lot about, and in fact, my research on homelessness was very much in that line of poverty. And that's a hard one to crack. I mean, I don't have an answer to that, but I will tell you that poverty is intertwined with a lot of other issues, education being one of them, drug and alcohol abuse being one of them, homelessness being one of them, health issues being one of them. You have all of these interrelated issues that lead one to poverty. Generational poverty is probably one of the biggest, the biggest factors. Like in Omaha, Nebraska, where I was living before Colombo, there are more millionaires per capita than any other city in the United States. It's the home of Warren Buffett and his great company, Berkshire Hathaway, probably the greatest living investor alive, right? Second richest man in the world. He has made many millionaires. And there are also 5 Fortune 500 companies there. But there are also more black children in poverty than any other city in the United States per capita. So you have this tale of two cities. On the one hand, the richest of rich, and on the other hand, the poorest of poor. And it's often generational poverty, and unfortunately that also plays into systemic racism. And for generations, African Americans, Black Americans have been in generational poverty, and that gets passed down, right? So the privileged become more privileged, and there's a great book by an author named Robert Putnam This is not his social capital theory. Well, it is in a way, actually, it's related. But it's called Our Kids. And it's talking about how basically rich educated people are marrying other rich educated people, and their kids are getting all these advantages. So they have private, music lessons, and they get to go on these trips, and they, have the best private education, and then they go to the best universities. And it's actually one thing that I would say Sri Lanka is a more meritocratous society in that free education is at the heart of giving everyone a chance. Now, these exams that are so much pressure on these kids, it's like your whole life is determined by that one day. But if you're good and you're a topper, it doesn't matter what your background is. You can come here and go to Kelaniya, right? And you can be a president of Sri Lanka. Like that is a reality now. And to me, sending that message instead of, is your daddy rich? Is your mama rich? What does that mean for your future, right? So I actually think that Sri Lanka has a lot of things going for it right now. And again, to the mindset, I hope that one thing you're taking away from this talk is that I am optimistic about the future of Sri Lanka. My wife moved here. I moved here in January, took this new job. My wife moved here in August. She just quit her job. She was 25 years as the music director and conductor of the Omaha area youth orchestras. So she's an orchestra conductor, probably one of the top 10 youth orchestra conductors in North America. And she gave up that career to come here and be with me in Sri Lanka. Our 23-year-old daughter has also just moved here. She just graduated from Oberlin College and Conservatory. She has dual degrees, a Bachelor of Music in Viola Performance. Viola is like a big violin. Viola Performance from the Conservatory, and Bachelor of Arts in Theoretical Mathematics from the college. And she has a grant to come here to Sri Lanka and learn from and collaborate with the greatest living contemporary artists across this island. So she's in this, at 23 years old, in this kind of cultural creative moment. It's great. My younger daughter is in London. She just finished her degree there in contemporary dance. So she wants to be a dancer and be a professional dancer for as long as her body lasts. So I am really excited about at least 3/4 of my family being here in Sri Lanka with me and making a commitment for the long term to be here. I hope that you make a commitment also to be here for the long term, and that you find ways to partner with me and with Fulbright going forward, because it is a life-changing opportunity. Thank you all very much for the time you've spent with me.
Thank you very much, Dr. Patrick, for that wonderful and insightful session. Now, to give away the token of appreciation for Dr. Patrick, I would like to invite the chairman of the University Grants Commissioner, Senior Professor Kapila Seneviratna, on stage. Thank you very much, sir. With that, we will be starting the technical sessions. This session has 5 abstracts listed for presentation. 10 minutes are allocated for the author to present their abstracts, and 5 minutes are allocated for the Q&A. When you have 2 minutes left, we will notify you by ringing a bell once and twice to indicate when the 10-minute presentation time has ended, and ring in 3 times to stop presenting. This session chair will then conduct the Q&A. Your cooperation in conducting the session productively is much appreciated. Now, let me briefly introduce the session chairs for this session. Dr. Thelma Abesinghe from Open University of Sri Lanka, and Dr. Tharindra Veerakorn from Department of Chemistry, University of Colombo. Madam, over to you.
Thank you. So after a thoughtful, provoking session, so we are coming for the scientific sessions. So in order to initiate this session, we have this presenter, Veerakorn Mudyansalage, Naomi Gloria Veerakorn. She will discuss about extraction of lipase enzymes from rice bran and rapid isolation of thermostable lipase by thermal denaturation of other proteins.
A very good afternoon to all of you who are present here, especially to the session panels as well, and my fellow presenters. I'm Naomi Veerakorn, and I will be presenting on the extraction of lipase enzymes from rice bran and rapid isolation of thermostable lipase by thermal denaturation of other proteins. So moving ahead, this is my outline in which I will give a brief introduction onto my research, followed by the research objectives, as well as the methodology that I've used, and the results, followed by the conclusions. So without further ado, I will be giving a brief introduction. So rice is known to be one of the main components that is being used in the human diet for almost half of the people around the world. And a byproduct of rice is rice bran, which is produced by the milling of rice. So, rice bran is known to have a short shelf life due to its rancidity that is resulting from the lipases that's present in rice bran. So in rice bran, there are 3 lipases that have been found, namely lipase 1, lipase 2, and the thermostable lipase. So I will be mainly focusing on the thermostable lipase, and thermostable lipase is being tested here in this research for its valuable use as a byproduct of rice bran, for its use in the detergent industry, for it to be used to remove lipid stains at high temperatures. So moving ahead, my research objectives were to extract protein from the rice bran and to extract lipases in rice bran, followed by the extraction and rapidly isolating the thermostable lipase from rice bran by thermal denaturation of other proteins, which is a method that we are testing, we are newly testing. So moving ahead to the methodology, rice bran extracted from the rice mill, from the milling of rice was taken and it was defatted using hexane. Afterwards, protein was extracted using 50 millimoles of tris HCl buffer at a pH of 8.0. It was then followed by the thermal denaturation of other proteins by rapidly isolating the thermostable lipase, and this was conducted at 2 different temperatures, 80 Celsius and 100 Celsius. So the concentration of the lipase was calculated by using a lipase activity assay, which is the phenyl acetate assay, by measuring its absorbance at 765 nanometers using a spectrophotometer, and the concentration was calculated, out of which the protein yield in gram per kilogram of dry weight of rice bran for thermostable lipase were calculated, and the results are shown here. So the protein yield at 80 Celsius is 3.05, and at 100 Celsius it was 2.7. So following up with my conclusions for the study, the results indicated the activity of lipase, and the results indicate the presence and the activity of thermostable lipase in rice bran at both temperatures of 80 Celsius and 100 Celsius. So extraction of and isolation of thermostable lipase in rice bran was successful, and hence it proved its worth and could be further tested for its potential use in the detergent industry, so which is the next step of this research that has to be tested for its potential use in the detergent industry at high temperatures, specifically to remove lipid stains. So I've come to the conclusion of my research, and I would like to acknowledge my supervisors, Professor Nimanti and Dr. Dulita Bekun, as well as my coordinator, Ms. Punkala, and laboratory assistants, Ms. Tashmi and Ms. Diniti. And also I would like to thank my college, British College of Applied Studies and Oxford Brookes University, and the coordinators there who were Dr. Mihira and Dr. Tarindra. Thank you very much.
Thank you, Naomi. Now the floor is open for questions. Okay, so when you are extracting the proteins, so how did you make sure that you are extracting only lipases, or how do you get rid of the other unnecessary proteins?
So, ma'am, the first method was to extract the protein. So that was done using a 50 millimole of tris HCl buffer, and a lipase activity assay was done for that. After getting the concentration of that, we further did a thermostable lipase assay, test for to extract the thermostable lipase separately. So it was done by heat crashing. So another part of it was after protein extraction, we also did a lipase test. We did differential fractionation to find whether lipase was present.
What was the yield of your experiment? Like after you getting all the proteins, what was the yield?
The yield was, for the thermostable lipase, it was 3.5 at 80 Celsius, and at 100 it was 2.07. 2.7.
So when you say rice bran, what is your variety? Did you specific for a variety or rice variety? Like there are many varieties, so depending on the variety, content of lipase can be vary, right?
Yes.
So what is the variety that you have chosen?
So the variety that was chosen was BG352, and this was taken because of its high, because it was being farmed in the local produce, like in a wide range.
And I think Dr. Thelma also asked the same question. So what is the technique that you have chose to separate the lipases, like lipase 1, 2, and thermostable lipase?
Yes, ma'am. So we did differential fractionation. Fractionation we used at 3 different salt concentrations.
Okay, so any techniques like ion exchange, size exclusion have you done?
Yes, we did size exclusion chromatography as well.
Now one more question. So now you have successfully isolated the lipase fraction, and yes, what you wanted is to develop as a detergent. Am I right?
Yes, ma'am.
So whenever whenever you wanted to, like, you are going to value added the product, so how economical is it? The product, if you developed.
I didn't get your question.
No, I mean that if you want to develop this as a product,
yes,
so whether it is cost effective, that's my question.
Yes. So, ma'am, we actually did a lot of testing, and we were able to maximize to get the maximum results. We maximized the procedures to get the highest yield we could in the least amount of money and the least amount of reagents and all the reactants and all.
Yeah, now there are many products available in the market.
Yes,
so did you like compare with those products available in market? If you develop a product from your one, is cost effective and economical, or did you see the feasibility study?
The feasibility is still being studied.
Okay.
Yes.
Good, good.
So this, I only isolated the thermostable lipase, and it's the research is still undergoing.
Okay. Thank you.
You're welcome.
Okay, thank you very much.
You're welcome.
Our next speaker is IP Madhushika. She will discuss on comparative study on physicochemical properties of wild and locally farmed bee honey in Sri Lanka.
Hello everyone. So my research was based on comparative study on physicochemical properties of wild and locally farmed bee honey in Sri Lanka. And I am WIPM De Silva, and I'm from Department of Chemistry, Faculty of Science, University of Colombo. And here is my outline. Introduction, research objectives, methodology, results and discussions, conclusions, acknowledgements, and references. So when we're talking about honey, what comes to your mind? It is a natural, sweet, viscous material produced by honeybees, either from nectar of blossoms or secretions of living plant parts. So during this process of honey production, sugary materials are broken down into simple sugars. Honey contains various nutrients that are good for human health. So because of that, there is a huge demand in local and international market for bee honey. So that some honey farmers tend to upgrade and increase their yield. So because of that, they do some wrong practices, and also due to some the sources of honey, the signature characteristics will be different between the locally farmed bee honey and wild bee honey. So here are my research objectives, and the aim of this study to determine the differences in major physicochemical parameters between wild and locally farmed bee honey in Sri Lanka, and ultimately to understand the potential of Sri Lankan bee honey for further research developments. And here is my methodology. A number of 7 samples were collected and numbered from each of 2 categories, and these samples were mixed by stirring and shaking, and then the pH, color, conductivity, moisture, ash, sugar, HMF, nitrogen, and acid-insoluble ash contents were studied. And all the analysis was done in triplicates. Color determination was simple, and the color was compared with the standard color scale, and determination of pH was done by preparing a 10% honey solution, and it was a direct measurement from the pH meter. And then the moisture content was determined by the refractometer, and the refractive index was taken, and there is a connection between the refractive index and the moisture content of bee honey. So there is a standard table, and by comparing with the standard table, the moisture content was determined and they expressed as % by mass. And determination of ash content was done by ignition of the sample at 600 centigrades in the muffle furnace, and by obtaining the weights, the weights were introduced to this equation and the ash content was determined. And the previously ash samples was taken and treated with 25 milliliters of concentrated HCl, and then it was filtered by slow filter paper, and then the filter paper was ignited in the muffled furnace, and the weights were taken, and the weights were applied to this acid insoluble ash content equation, and it was determined. And then the specific conductivity is also a direct measurement for 10% honey solution. And HMF content was determined according to the AOAC official method 980.23, and the absorbance of wavelengths at 284 nanometers and 380 nanometers were taken, and the readings were applied to this equation, and the hydroxymethyl furfural content was determined. And determination of total sugars was done by Lane and Nynan method, and by this equation, the final sugar content value was taken, and the sugar value was expressed as percent by mass, and nitrogen content was determined by Gel-Dahl method, and here is the equation to determine the nitrogen content, and as you all know, Gel-Dahl method involves digestion, distillation, and titration. The titration reading was taken to determine the nitrogen content. And statistical testing of results were done by using the SPSS 2001 software. Moving on to my results and discussions, when we are talking about the color analysis, the color of honey samples falls within the range from light yellow to amber. And according to the results, many wild honey samples were ranged from amber to light amber, while the color of cultivated honey was ranged from water white to light amber. And wild honey was frequently darker due to the presence of phenolic compounds. And moisture content, when we are talking about the moisture content, as you all know, moisture content is a criteria for determine the quality of a food sample. So it determines the stability against microorganisms and resistant to the spoilage. And in this analysis, the locally farmed bee honey samples showed average value of 22.6, while it's for wild bee honey became 20.3. And the results supported the previous findings. Statistically, there is a significant difference between the two categories. And when we are talking about sugar content also determines the overall quality of honey, and the average sugar content for locally farmed bee honey was 73.4, while it's for wild bee honey was 75.2. And there was significant difference between the two categories. And when we're talking about the pH value, here are the pH values we obtained for the analysis. And when we are talking about the conductivity measurements, average conductivity for locally farmed honey samples were 0.36 millisieverts per centimeter, while it is for wild honey was 0.43 millisieverts per centimeter. And there was a significant difference between the two categories. And when we are talking about the ash content, the average ash content for locally cultivated honey samples was 0.08, while it is for wild honey 0.03. And as you all know, ash content reflects the amount of mineral content in honey. And there is no significant difference between two categories. When we are talking about HMF content, the locally farmed bee honey samples had the average content of 8.49 milligrams per kilogram, while it was for 0.08 milligrams per kilogram in wild bee honey. So there was a significant difference between the two categories. And when we are talking about the acid insoluble ash content, the average acid insoluble ash content for locally farmed bee honey samples was 0.0028, while it is for wild honey samples was 0.0014. There was no significant difference. And when we are talking about nitrogen, average nitrogen content in locally farmed honey was almost zero, while it was for wild bee honey was 0.1. And there was a significant difference between the two categories. And here is the conclusion. According to the study, moisture, pH value, conductivity, HMF content, and nitrogen content showed a significant difference between the two categories, but total sugar and ash contents showed no significant difference. And all readings met the Sri Lankan standard for bee honey. And here are my acknowledgements to the Department of Chemistry, Faculty of Science, University of Colombo for providing necessary guidance, and also for Faculty of Graduate Studies, University of Kelaniya for organizing this valuable platform to present our researchers, ultimately my closest circle for their kind support. And here are the references. So thank you very much, everyone.
Thank you. So now it's time for questions.
Now, one of the main problem that we face in Sri Lanka is adulteration. How are we going to detect whether the bee honey is adulterated or not, whatever available in the market?
By determining the HMF content. And also, I think Industrial Technology Institute has developed a solution or test solution for that. And when we add that solution, it turns into red.
What is that solution?
I can't remember. It's their new innovation.
HMF
I think.
Is why we looked at HMF?
To get some kind of an idea about the adulteration.
No, it is the toxicity you look at.
Sorry?
It is the toxicity. HMF, if it is, it's a certain level, if it is above that, then it is toxic. So basically the sugar content, I guess, like fructose, glucose, and sucrose, those are the sugars that are available. And I think that ITIA have developed a test kit to show that whether those proportions are available.
Yeah.
That is, I think, that is what is important because we need to standardize it and get the good quality bee honey.
Yes, madam.
Okay.
So I have one question. So in the methodology you have mentioned, you have collected these bee honey samples from 7 different places. So have you restricted to a certain area in the country, or have you collected in different areas of the country and mixed them?
No, we collected from different areas of the country.
So depending on the geographical location of these bee samples, do you think that there can be any variation? I mean, especially there can be variations in the adulteration as well. According to my point of view, what is your view about that?
Yes, I also think so, due to the geographical origin and also due to the geographical origin, the plant sources and the content in those chemical content of the plant sources will be differ. So I think your idea is good, madam.
So in your samples, the wild type of not the industrially grown one, the other samples were, I think, be darker in color. So
no, wild samples were darker in color.
So that was darker in color. So why do you think it has happened?
Due to the presence of phenolic compounds. More phenolic compounds are there in wild samples than the locally farmed samples. And also maybe due to the, I think, due to the mix in with corn syrups and sugar syrups, the color will be decreased in the locally farmed bee honey samples. But I think in the wild bee honey samples, the color was increased and color was darker due to the presence of phenolic compounds.
So you have not tested that, like the total phenolic components or the amount you have not tested?
No, I haven't, madam.
Okay, thank you very much. So we have our next speaker, MM Piumi Madhubashini, on development and evaluation of a quinosoline derivative as a urease inhibitor for reducing nitrogen loss in agricultural soil.
Good afternoon, everyone. I am Piumi. My research is development and evaluation of quinosolinone derivative as a urease inhibitor for reducing nitrogen losses in agricultural soil. First of all, this is the outline of the presentation: introduction, research objectives, methodology, research and discussion, conclusion, acknowledgement and references. First of all, we will move on the background of the research. As you all know, urea fertilizer is nitrogen-rich synthetic fertilizer used worldwide to boost plant growth. When urea applied to soil, urea reacts with water, producing ammonia and carbon dioxide through a process called urea hydrolysis, which is catalyzed by the urease enzyme. Soil microbes then further break down the ammonia, converting into ammonium ions and other nitrogen forms such as nitrate, nitrite, nitrogen oxide and nitrogen dioxide. However, not all nitrogen form of urea is absorbed by plant. Some of it lost through various processes, such as ammonia volatilization, leaching, inefficient nitrogen uses, and denitrification. To address these nitrogen losses from urea, urease inhibitors are commonly used to slow down the hydrolysis process. The inhibitors can apply directly or combine with urea. In my research, I focused on synthesized cost-effective inhibitor to reduce nitrogen losses. In this study, I selected quinosolinone derivative because it exhibits excellent antimicrobial, antifungal activities while being low in toxicity to plant. My objective of the research was to conduct computational analysis of 3-amino 2-phenyl quinosolinone interact with Bacillus pasteur urease enzyme and to synthesize 3-amino 2-phenyl quinosolinone to confirm the structure of all synthesized compound using NMR and FTIR analysis. Then to evaluate the inhibitory activity of 3-amino 2-phenyl quinosolinone using urease assay. Before the synthesis of inhibitor, it is important to understand how it interacts with urease enzyme. According to the literature, there are various soil bacteria that contain urease enzyme. Among these bacteria, Bacillus pasteur is a common soil bacteria found in various soil conditions. I select Bacillus pasteur for docking study because it is well characterized regarding its urease activity and shows good interaction with urease enzyme. When urea fertilizer applied to soil, the inhibitor bind to the active site of urease enzyme and it is blocked the urease. As a result, urea remaining in the soil for a long period and is released slowly. I compare the binding affinity of the synthesized inhibitor thiourea, which is commonly used as a reference molecule for urease inhibitors. I used Gold 530 version, Discovery Studio Visualizer and Avogadro software for docking study. Next, move on the synthesized path. The synthesized pathway was carried out using anthracic acid as a starting material. Anthracic acid was treated with benzyl chloride in pyridine along with sodium hypochlorite to produce 2-phenyl 4H benzoxacinon. The product was refuxed with hydrogen monohydrate and heated between 200 Celsius to 250 Celsius degree for 25 minutes to obtain 3-amino 2-phenyl quinosolinol. This is the target inhibitor. Resulting compound was pre-crystallized using ethanol. The structure of the product was characterized by FTIR, proton NMR and carbon NMR. Next, move on the results and discussion part. The first table shows the molecular docking results. You can see the binding affinity of the synthesized inhibitor is greater than that of reference molecule standard thiourea. The next part is the urease assay results. The urease assay was performed on standard thiourea and synthetic inhibitor. The same reaction applicable in this process. If the inhibitor successfully blocked the urease enzyme, no ammonia produced. That means no color development. The first curve shows the percentage inhibition was consideration of standard inhibitor thiourea. You can see the IC50 value is 14.5 ppm, and the second curve shows the percentage inhibition was consideration of synthesized inhibitor. The IC50 value was 17.5, which is close to each other. That means the synthesized inhibitor is nearly as effective as the thiourea. The conclusion. Actually, the synthesized inhibitor is combined with urea before applied to soil. I did a urease assay for several ratios such as the urea and inhibitor mixture 1 to 0.5, 1 to 0.05, 1 to 0.005. In this ratio, no color development occur. That means inhibit the all urease enzyme. Then I moved to 250 to 1 ratio. In this ratio, I was observed the color development. That means it is the optimal ratio. Then the research finding indicate that only a small quantity of synthesized inhibitor was required to achieve effective inhibition. And acknowledgement, I would like to thank University of Kelaniya and my supervisor. These are some references and thank you.
Thank you. Now the floor is open for questions. I have a question. In your binding studies, you have noted that the active site will be bound to this particular synthesized compound. So is it like binding completely irreversible, or like is it a reversible binding?
Not completely. The main binding site are nickel atoms, histidine, lysine. Some docking points, nickel atoms are binding, but not completely binding, as the binding effect is 50% like.
So there's a possibility of reversing the reaction.
So in the graph that you have shown for the comparison, so what is the synthesized inhibitory product that you have synthesized, that with the increasement of the concentration, the inhibition shows little bit reduction, right, when compared to the standard inhibitor that you have used? What can be the reason for that?
Increase in concentration, inhibition is increasing.
Yeah, but after a certain point, like a parabolic curve.
I found IC50 value.
Yeah,
That's
IC50 during that range, I can see it as a straight line, but once the concentration, I can't remember the exact, it shows like
constant.
Yeah, constant curve. Do you have any idea about the reason?
In this concentration is the maximum concentration, the constant of the inhibition. Then I focus on the 50% inhibition.
I see. Okay.
One more thing, that just a clarification. Now, usually you use usual inhibitor is the thiourea, but now you have used the aromatic compound. Why? On what reason that you selected that type of compound as a
first of all,
inhibitor?
First of all, I did the molecular docking. The molecular docking results, the Bacillus pasteuris is my target urease enzyme. This enzyme more inhibition shows this inhibitor, quinosolidine.
Because structurally both are not similar. The thiourea structure and quinoline structures are completely
NH2
different.
group is very highly bind with the urease enzyme for Bacillus pasteuris.
Okay.
Since we have no more questions, thank you. Our next speaker is MTS Uresha on extending the shelf life of deodorized Aluritis mollucana oil using phenolic extracts from coconut pairing residue.
Good afternoon, everybody. My research is extending the shelf life of deodorized Aluritis mollucana thelkakuna oil using phenolic extract from coconut pairing residue. So the outline of the presentation is introduction, research objectives, methodology, results and discussion, and conclusions and references. So let's jump into the question, what is thelkakuna? Actually, thelkakuna, its scientific name is Aluritis mollucana. It is a evergreen plant which is basically found in the wet zone of the Sri Lanka. And it is a medium-sized tree which grows up to 20 meters in height. The kakuna nuts are covered with a hard shell. So it is the kernel that we use to extract oil. And the kakuna nuts are also in English it is referred as candle nut, candleberry, varnish tree, and Indian walnut. So there was one specific word in the topic, that is CPRE. So what is CPRE? CPRE stands for coconut pairing residue. Coconut pairing residue is the leftover material after coconut oil is being extracted from the coconut material from the kernel. So the pairing means for the brownish out layer of the coconut kernel, and it's also known as the pairing. The phenolic extract of the coconut pairing residue was used for the study. So then the important question is why we use thelkakuna. Thelkakuna is a highly underutilized plant in Sri Lanka, but it has a great potential to produce a beneficial oil. It is loaded with omega fatty acids, antioxidants, and it's only currently being used to light oil lamps. And so the very word tel means Sinhala, it means oil. So that implies that it is loaded with oil, and also in the English word, this candle means it has been used to illumination purposes. So the main drawback of the telkakuna oil that it has a natural pungent odor, so that reduces palatability. That is why it needs deodorization. So these are the research objectives: to deodorize the telkakuna oil and to preserve the antioxidant activity to extend the shelf life. So this was the methodology I followed. Then these cacao nuts were purchased from the local market, and they were cracked open, and the nuts kernels and the shells were separated, and the kernels were dried and made into a powder. Then this powder was subjected to extraction, and next in the rotary evaporation, and the telkakuna oil was obtained. So following treatments were done. So the 3 samples was crude oil, stripped oil, and stripped oil plus CPRA. Stripping was done using an activated alumina column. So following characterizations were done: fatty acid profile, shelf life test, alpha tocopherol and beta carotene analysis, and total phenolic content and antioxidant activity, DPPH assay. And besides that, oil yield was also calculated and peroxide value also monitored. So in the result, let's move to the results and discussion. So in the figure 1, it shows the HPLC chromatogram of the telkakuna oil. So you can see 2 peaks. The first peak is alpha tocopherol, which means vitamin E, and the second peak is beta carotene. So the values were quantified as 34.68 for alpha tocopherol and beta carotene 4.12. So the total phenolic content and the antioxidant activity were assessed. So in the table 2, these values have been are shown. So for the 3 samples, crude oil, stripped oil, and stripped oil plus CPRE, as you can see, there was no significant difference in DPPH radical scavenging activity between crude oil and stripped oil plus CPR, which means that the antioxidant activity and total phenol content are same in the crude oil and the stripped oil plus CPR. So the fatty acid profile of the cacao oil was monitored. There are basically 4 fatty acids. There are palmitic acid, oleic acid, linoleic acid, and linolenic acid. So the percentages were observed as 5.39 and 28.16, which is being second highest, and 45.84 and 20.16 for the respectively. And as you can see, linoleic acid is an omega-6 fatty acid, and linolenic acid is omega-3 fatty acid. So omega-6 to omega-3 ratio here is 2 to 1, and this is this omega-6 to and omega-3 ratio is a buzzword in nutrient industry, and it's highly important. So the favorable ratio is falls between 1 to 1 and 4 to 1. So in here, in telkakuna, it is 2 to 1, which means telkakuna is it has a better omega-6 to omega-3 ratio. So the oxidative stability was monitored using random test. As you can see, for the oxidative stability of the stripped oil plus CPRE was 27.01 hours, whereas the crude oil exhibited stability only for 3.0 hours at 120 Celsius. So these are the conclusions. So the oil seems to have a favorable balance between omega-6 and omega-3 fatty acid, which is important for overall well-being. And CPRE could be a compound or ingredient that helps to restore or replace the missing these antioxidant and ensuring the oil retains its protective properties against the oxidation. After the deodorization process, oils usually use their antioxidants, and they are more susceptible to spoilage and rancidity. So CPRE can replace that activity and extend the shelf life of telkakuna oil. And stripping process deodorize the oil while enhancing its palatability. These are my references. And thank you.
Thank you. Now it's time for questions.
fatty acid profile from GC?
From the gas chromatography, madam.
Yeah, how did you prepare them?
I prepared fatty acyl methyl esters and using that I inserted it to the gas chromatography.
What method you use to use for the identification?
That means, you are referring, madam, that in order to identify the peaks and the so we ran with the standard and identified the peaks.
Okay, okay. You didn't do GCMS?
No, madam, we didn't do that.
And also, when you do the testing with rancimate, did you what is the standard you use to? Did you use a standard to compare the shelf life with your sample? Did you use it?
So, madam, we basically we just ran these samples because there was no kind of standard. I mean, the different oils have different shelf lives. No, so, madam, we didn't use a standard, but
you didn't do a comparison, right?
No, madam, we didn't ran these three
you just
samples.
looked at for this particular oil.
So, how the activity the reference was the crude oil. So, what is the performance of this stripped oil plus CPRE reference to crude oil.
What is the purpose of increasing the shelf life of this oil? Like, are you planning to use it for some economical?
Yeah, madam, this can be used to produce economically important products like soap and cosmetic materials.
So, when we compare coconut oil and this cacuna oil, so instead of using coconut oil itself, so do you think using this cacuna oil to produce this soap and all detergents, will it be cost effective?
Yeah, but definitely it will be cost effective, but I agree with the point that it is, the popularity of this plant is quite low when compared with the coconut oil, but when in the production process, definitely this will be cost effective.
I have one clarification in your methodology. So did you extract the oil itself, or did you take the fraction that goes to the hexane? So it was not clear to me, like whether you got the crude oil just extracted from the kernels, or you have extracted whatever the fraction go to hexane. For analysis.
From the rotary Soxhlet extraction. So what is extracted? In the Soxhlet extraction, this oil get dissolved into the N-hexane, and so in the rotary evaporation, so you can evaporate the solvent and you can get the crude. No, madam. So
why won't you, like, you can just get the crude oil, like what we get coconut oil is like you get the oil itself and then do the analysis. So what is the purpose of having
madam,
this extraction?
there is some difficulty. It is better if you can go for the mechanical process directly, but in order to go for the mechanical process, you need a quite large amount of cacuna nuts. So we were basically trying to get rid of this pungent odor in the very basic step of extraction, but it was also successful. But in the natural process, when the intensity has definitely been reduced.
So after all your processes, this pungent odor like completely went away, or like were you able to measure it somehow?
No, that means, madam, like a sensory evaluation.
Yes, exactly.
Yeah, so I agree with your point, madam. I didn't do a sensory evaluation. That this still should be completed. Yeah. Thank you, madam.
Okay, thank you. So with that, we are moving to our final. Yes. Okay, sorry.
Hello. You said that cacuna oil can be used as a soap and other oil manufacturing. When you go the industrial manufacturing production, So you said coconut oil is not popular or less amount of shells we can get. So how you transfer into industrial application? And why don't you try when instead of adding coconut CPR into that, but its shelf life is poor, you can add some more vitamin E on there. You will extend the vitamin shelf life of the oil.
Yeah, vitamin E is only one antioxidant, but there are several other, there is possibility of containing several more antioxidant in the coconut oil. So rather than basically for just focusing for one antioxidant, that is why that is CPRE contain a lot of antioxidants and rather than just one antioxidant. That's why we were basically focusing on CPR.
So you can add that in control as well. If we are externally, so we nowadays more develop and derivatives, vitamin E derivatives are in the market. It can use as a shelf life of coconut. It's a coconut oil itself. So many companies are using that vitamin E, newly derived vitamin E. I have heard some cosmetic manufacturer. So you can look into that. And so in the saffronification process, when we made a soap, with the saffronification process, that order could be changed, I think. So you need not deodorize to that oil.
Yeah.
So deodorizing the oil is not. If you are going to make soap, deodorizing is not important, I think.
Yeah, not soap. There's also have some pharmaceutical value also.
Okay.
Like we use this olive oil and everything. We also this oil also
so you have
can
to
be
mention
used.
that.
Okay, thank you.
Thank you.
Yeah.
Thank you. So we have our last presenter for this session, SU Kankanamge, on extraction of starch from Chinese potato yams and characterizing by chemical and physical modifications as novel alternative pharmaceutical excipients.
Distinguished session chairs and my dear fellow presenters, good evening everyone. I am Sama Kankanamge and my study is about the extraction of starch from Solanaestemon rodentifolius yams and characterizing them by chemical and physical modification as novel alternative pharmaceutical excipients. This is the outline of my presentation. As we all know, starch is one of the main excipients used in manufacturing of tablets and capsules. Nowadays, there's a trend in use of modified starches due to their several benefits. So in this study, I have used acetylation as the chemical modification technique and pre-gelatinization as the physical modification technique. And I use the starch source as the Solanaestemon rodentifolius. It is locally named as innuller, which contain over 60% of starch. The significance behind this study, most of the commercial starches are being imported to the local pharma industry, facing many problems like high importation costs, sudden shortages, late deliveries, and also recently scientists have found maize starches there's a potential to causing allergies to the patient, as well as some issues encountered in maize starches like poor flowability and poor compressibility. However, there's a possibility to produce the required starches utilizing the natural starch resources available within the country. Thereby, we can reduce the importation cost and cut down the total manufacturing cost of the pharmaceutical. And finally, we can deduct the price of the medicine in the local market, especially during this kind of economic crisis situation. And also, there's no detailed studies have been carried out to analyze these modified starches of this innuller as a pharmaceutical excipient. Therefore, this study aimed to extract starch from these yams, innuller yams, and convert them to acetylated and pre-gelatinized starches. and characterize their physicochemical properties compared to the commercial starches, maize starch, BP. Moving to the methodology, commercial starches, maize starch, BP was collected from State Pharmaceutical Manufacturing Corporation in Sri Lanka, and yams were collected from Palmadul area in Sabaragamuwa province, Sri Lanka. And we extract the starch from these innal yams by mixing the powdered yams with the distilled water in 1:2 ratio, and that slurry was kept for 3 hours, and then it was blended and filtered. Then the filtrate was kept for 48 hours. After 48 hours, the sediment was separated and dried at 40 Celsius degree. Thereafter, the dried starches was treated with sodium hydroxide, acetic anhydride, and HCl to obtain acetylated yam starch, and pre-gelatinized starch was obtained by heating the starch slurry to 80 Celsius degree for 15 minutes with mixing. Thereafter, we did the iodine test and also the organoleptic properties evaluation. We determined the percentage of acetyl group in the acetylated innal starch and the physiochemical properties like pH, bulk, tap and true densities, angle of repose, Hoshner ratio, Karl's index, hydration capacity, moisture absorption capacity, swelling power, clarity and viscosity were assessed. In addition, scanning electron microscopic images, X-ray diffraction pattern, and IR spectra of all these starches were analyzed. Finally, we did the t-test to compare those data. According to our results and discussion, all these starches type provided positive results for the iodine test, giving blue color appearance at the end. And the percentage of acetyl group in the acetylated innal starch was 1.26%, which is complied with the Food and Drug Administration standard, which is not exceeding 2.5%. And according to the organoleptic evaluation, maize starch, BP, native, and acetylated innal starch had smooth, no-shine texture, while pre-gelatinized innal starch had rough, shiny texture. And maize starch, BP had matte white color, native and acetylated innal starch had light yellow color, and pre-gelatinized yam starch had light brown color. Considering the scanning electron microscopic images, maize starch BP had polyhedral shaped starch granules, and native and acetylated innal starch had dome shaped starch granules, and pre-gelatinized starch had flat shaped starch granules. According to the XRD pattern, maize starch BP, native and the acetylated innal starch had similar XRD pattern, giving the peak in 15, 17, 18, and 23rd position at the 2 theta angles, but in pre-gelatinized starches, the XRD pattern was diversified by the pre-gelatinization. According to the FTR spectra, all the 4 spectra provided similar peak formation around 3000 and the 1000 range, indicating the hydroxyl group and the glycosidic bonds respectively as present in the basic structure of starch. However, we could observe a new peak formation around 1727 in acetylated innal starch, assuring the acetylation. But we couldn't find any new peak formation in the pre-gelatinized starch. This table shows you the physiochemical profiles of the native and modified innal starches compared to the maize starch BP. You can see most of the physiochemical parameters are significantly different with the maize starch BP, and some of the parameters like angle of repose, Karl's index, Hoshner ratio have been reduced in modified starches compared to the native and maize starch BP. Accordingly, we can conclude that acetylation and pre-gelatinization exhibited significant changes in the physiochemical parameters of these innal starches with the improved flowability, swelling ability, and compressibility compared to the commercial starch, maize starch, BP, and its native form. These improved properties add value to these acetylated and pre-gelatinized innal starch, and we can encourage local pharma industry to aim these novel starches as excipient rather utilizing pricey commercial starches. Therefore, we recommend for these studies to assess the suitability of these modified starches as a binder, filler, disintegrant in formulated suitable pharmaceutical dosage form, and also we can attempt other modification techniques and other natural starch resources available in Sri Lanka. Finally, I would like to acknowledge SPMC and the Instrument Center of the University of Sri Jayawardhanapura, Department of Material Science, University of Moratuwa, University of Ratmalana and the Bandanayaka Memorial Ayurveda Research Institute, Navinda, and the Kothalavala Defense University for providing the raw materials and the facilities, and also my beloved supervisors for their kind guidance. This is the part of the reference I referred in.
An interesting study. Were you able to characterize the starch you have extracted based on the stability? Like
Yeah. We tested for the microbial, total aerobic microbial content and also yeast content.
So like it's comparatively stable?
There's microorganism level is comparatively high in yam starch compared to the commercial starches.
And also like now you have like future part of this one is like you are going to incorporate it into capsules as a binder and all. So how soluble is this starch?
Yeah, solubility index is also high. I couldn't include it here compared to the maize starch BP.
So why do you specifically select this Chinese potato species? Is there any reason for that?
Yes, madam, because it's highly available and easy to cultivate in Sri Lanka, and also considering its high yield and the starch content.
Okay, so since no more questions,
Thank
let's
you very
thank the
much.
speaker again.
Thank you very much, and that marks the end of the technical session. Now I would like to invite the chairs of the session to award the certificates to the presenters. Presenters, please come on to the stage as I read out your names. Naomi Gloria Viracon. IP Madhushika. MM Pumi Madhubashini. MTS Uresha. SU Kankanamge. Thank you very much. We would like to convey our thanks to the presenting authors for their submissions and the many interesting concepts, ideas, and applications that were set out in different areas. We trust that based on your findings presented here and the discussions, you will be able to improve and further refine your research study. On behalf of the organizing team, we would like to thank our session chairs, Dr. Thelma Basinger and Dr. Tharinda Veerakorn, for providing their professional input to the authors and the audience here. And I hope that the discussions will help the authors to refine their work further. Now, to give out the token of appreciation to our session chairs, I would like to invite one of the session coordinators to deliver the token of appreciation. Thank you very much, ladies and gentlemen. That concludes the session, and we will meet again soon at 3:15 for the next session. Thank you very much.
Good day, good afternoon. Welcome all of you for the last session of IPRC conference. So we will start the session now. The first okay.
Good evening, ladies and gentlemen. On behalf of the Organizing Committee, Faculty of Graduate Studies, University of California, we would like to welcome you to the 24th International Postgraduate Research Conference. This session has 7 abstracts listed for presentation. 10 minutes are allocated for the author to present their abstract, and 5 minutes are allocated for the question and answers. When you have 2 minutes left, we will notify you by ringing the bell once and twice to indicate when the 10-minute presentation time has ended, and ringing 3 times to stop presenting. The session chair will then conduct the Q&A. Your cooperation in the conducting session productively is much appreciated. It is now my pleasure to briefly introduce the session chairs, Professor Priyani Parnagama from Department of Chemistry, University of Kelaniya, and Dr. Chamila Kadigamuwa from Department of Chemistry, University of Kelaniya. We would now like to hand over the session to Professor Priyani Parnagama and Dr. Chamila Kadigamuwa from Department of Chemistry, University of Kelaniya. Over to you, sir, madam.
Thank you very much. So we are going to start the last session of the 24th International Postgraduate Research Conference, IPRC 2024. I wish to invite the first presenter of this session, Ms. K.A. Aruni Alvis. And title of the presentation is The Preliminary Investigation of Shara Powder of Erythrina variegata used in Ayurveda Treasure. Over to you.
Good afternoon, dear all. I am Aruni Alves from Institute of Chemistry, Ceylon, College of Chemical Sciences. I am here to share my findings on the topic preliminary investigation of Shara powder of Erythrina variegata used in Ayurveda treasure. Shara is an Ayurvedic remedy that used to treat chronic non-healing wounds, fistula in ano and hemorrhoids. Shara is an alkaline substance and it's prepared by water-soluble ash of dried plant through special procedure mentioned by Acharya Sushruta Samhita, mentioned in the Sushruta Sutra 11th chapter. There are 3 main types of Shara according to the potency: Tikshana, Madhya and Mrudu. There are 24 plants mentioned in the text, and we have chosen the plant called Erythrina variegata, Erabadu, for the research. Since there are no scientific evaluations done under the Erythrina variegata Shara and its bioactivity like antioxidant, anti-inflammatory, antimicrobial properties, as well as it's used in Ayurveda for treatment in cuts and wounds and also bacterial infections. As the research objectives, Shara powder of Erythrina variegata have been prepared using systematic methodology started in the Ayurveda text for the investigation of the standard quality parameters of the Shara powders and evaluation of the bioactivity of the Shara prepared by using standard bioassays. Here you can see the methodology for the preparation of Shara powders. I have collected 20 kilograms of raw plant parts and shade dried for like 1 month, and the plant parts was burned in the outside and the ash was collected. 7.75 kilograms was observed, and the collected ash was mixed with 6 times of water, and the supernatant fluid was filtered 21 times using a cotton cloth until tea color observed. Then filtrate was boiled and evaporate the water. Then the powder was taken. When preparing Tikshana Shara, we have used
conic shells, and Chitraka, Plampaka Salonicus for the enhancement of the pH value. When preparing Madhyashara, we have used only the conch shell. When preparing Mrudashara, we haven't added any Chitraka or conch shell because the highest pH value for Tikshanashara, second for Madhyashara, and lowest pH for Mrudashara. Here you can see 10 and 11 pictures, Mrudushara and Tikshanashara, the prepared ones. Then the efficacy was evaluated using the total ash value, loss on brine, acid-insoluble ash and water-soluble ash, and also pH test was done for Tikshana and Mrudashara. As you can see, 10.98 for Tikshanashara and 10.29 for Mrudashara. And also, when it talk about total ash value, Tikshanashara was showed highest ash value with compared to Mrudashara. And loss on brine is, when talk about loss on brine, Mrudashara shows highest value than Tikshanashara. And also, FTIR has done for identification of functional groups. Mainly, it shows CO single bond stretching and OH stretching in the Erabudushara. And we have done DPPH assay using DPPH assay for antioxidant test. And here you can see the graph. We have used a standard as a standard BHT standard. So Tikshanashara shows the highest EC50 value with compared to Mrudashara. Then we have done anti-inflammatory assay according to the HRBC membrane stabilization method. And here you can see the graph and the values we observed. Here, Mrudashara shows highest inflammatory activity with compared to Tikshanashara. And also, we have done antibacterial assay according to the well diffusion method, and amoxicillin was used as the standard. Here you can see the inhibition observed for the Streptococcus pyogenes, E. coli, Staphylococcus aureus, and Proteus mirabilis. Okay, let's come to the conclusions. According to the findings, Erythrina variegata, Shara powders are significant valuable product because of its antibacterial, antioxidant, and anti-inflammatory activation. This can be further developed by enhancing the quality by applying advanced formulation methods and evaluation methods in order to commercialize. We will be thinking to develop a Shara-containing plaster in the future because this Shara is already used to wound healing properties in Ayurveda sector. So we will be thinking to develop a Shara-containing plaster in the future. Acknowledgements. Firstly, I would like to thank my principal supervisor, Professor Priyani Paranagama, my co-supervisors, Dr. Peshara Karyavasam and Dr. Anura Vikramanayaka from Indigenous Medicine, University of Colombo. Here are the references that I used to for my research, and thank you everyone.
Okay, thank you, Ms. Alice. It's a nice work you have done. However, there are some things that I want to know, to get from you. Now, when you collect the samples, how did you collect the samples?
I have to cut off 20 kilograms of Erebadu and I have to shade dry.
That, from where you get?
Minongod area.
How many plants?
It's like 20 kilograms or I think 3 to 4 plants. Yeah, huge amount.
Is that only one species in Sri Lanka or many species? That,
yeah. This is called, I think this is Yak Erebadu. There are so many variations, so this is Yak Erebadu I used.
Okay, thank you. And you use the whole plant?
Yes,
materials.
all the plant we have to use because that is how studied in the Ayurveda text.
Okay, is that how the Ayurveda do?
Yeah.
Take whole.
They actually they are using Shara, but they haven't done anything for Erebadu Shara.
Okay.
That's why we use that.
Then why they are doing ashing, not just extracting? What's the
Ash? Actually, with the ash.
Is that how they do? What's the reason for doing the ashing?
Because there are so many antimicrobial properties, antioxidant properties in the Shara, so that is how they are treating, making Shara actually. This is
According
how.
to my knowledge, I think when you ash it, they will lose the property.
Not only organic antioxidants are there, no, inorganic antioxidants also there. So I think it will show the result.
Okay. Because we are also doing same kind of things and we just extract them, but we are not ashing them, right? And also that's good. And then good to know that, right? And also these compounds are like that when you make the shara that become a brown color, right? Then when you do the DPPH assay or those are colorimetric methods, then is there any color interference for the assay?
Actually, the it was an ash color powder, and I used methanol extraction for the process. So nothing happened with the color.
Or you had the control that you had the something control, right? Methanol as the control, that zero, and then you zero to that, and then without DPPH assay, DPPH. Did you get a control that only the Shara?
No, I use BHT standard for the reference.
Okay, then don't you need the subtract? That is another control, I think. Blank, yeah.
No, I didn't use like that.
Okay.
I compared with the standard, BHT standard.
Okay.
Color difference.
Yeah. Okay, that's what I want to know. Okay. Yeah. Okay, then that's all I have. Thank you very much and nice presentation, and I hope you will make that bandage or something. Okay.
Thank you, sir.
Okay, thank you. We'll move to the next presentation. That's from Ms. Mohammed Harris F. Rihana. The presentation title of the presentation is Phytochemical Analysis and Mosquito Repellency Evaluation of Ocimum santum. Madhurutala.
Good evening, everyone. My name is Rihana Harris from Faculty of Applied Science from University of Sri Jayawardenepura. Today, I am going to discuss about my research project. My research heading is Phytochemical Analysis and Mosquito Repellency Evaluation of Ocimum sanctum. These are the outline of my presentation. Ocimum sanctum, commonly known as holy basil or tulsi. It is an aromatic perennial plant in the family Lamiaceae. It typically grows to about 1 to 2 feet in height. and it has a strong, pleasant aroma. The leaves are usually green leaves. Some varieties have purple leaves. Osmum sanctum plant has a rich phytochemical profile, such as alkaloid, phenol, flavonoid, glycoside, tannin, and steroids. And it has a wide range of pharmacological activities and medicinal uses, such as anti-cancer activity, antimicrobial and antifungal activity, and anti-inflammatory and antioxidant activity. And another special property is mosquito repellent property. We all know today world facing various kind of mosquito-borne diseases such as dengue, Zika virus, etc. It is continued to be major public health issue, especially in tropical regions. It necessitating the exploration of safer and environmentally sustainable repellent solution. In this context, in light of these concerns, this research investigates the phytochemical composition of Osmum sanctum and evaluate its efficacy as a natural mosquito repellent product. Then let's look about the research objectives. Our primary objective are to analyze the phytochemical profile of Osmum sanctum and assess its repellent effectiveness, and ultimately develop a natural sustainable repellent balm that could offer an alternative to synthetic repellent. Now let's look about the methodology part of the research. First of all, plant materials were collected from the Bandanayaka Memorial Ayurvedic Research Institute. Then herbarium sheet was prepared and it was authenticated by the herbarium of Bandanayaka Memorial Ayurvedic Research Institute. Then collected leaves were ground into the fine powder. Then methanolic extract and aqueous extract was prepared. Then phytochemical screening tests were conducted for both solutions. Then essential oil was extracted from the fresh leaves of Osmum sanctum via hydrodistillation process. At the same time, Aedes mosquitoes were reared in the insectary. Then mosquito repellent wooden box was prepared. It is 1 meter length. Then mosquito repellent box was separated into 2 equal parts using a middle wall. This middle wall containing a hole that 10 centimeter diameter. Then essential oil was placed in one part of the box. Then mosquitoes were introduced into that part. Then repelled mosquitoes were counted and recorded. Then mosquito repellent balm was developed using the essential oil and other ingredients such as beeswax, base oil, glycerin, aloe vera gel, and emulsifying oil and preservatives. Now let's look about the results and discussion part of this research. This table shows the summary of phytochemical analysis part. This table represents the phytochemical screening results of the aqueous and methanol extract of leaf part of Osmum sanctum. According to this table, alkaloid, glycoside, tannin, steroids were present in the methanol extract of leaf part of Osmum sanctum. And alkaloid, flavonoid, glycoside, tannin, steroids, phenol were present in the aqueous extract of leaf part of Osmum sanctum. These are the results of mosquito repellent evaluation part. The study was designed to evaluate the efficacy of 100% Osmum sanctum essential oil over 3 trials using water as a control. Results of water control test is shown in figure 1. In here, blue color line is represent the treated area, and red color line is represent the untreated area. In treated area, there is initially high amount of mosquitoes. In untreated area, there is none of any mosquitoes. Then during the observation period, mosquitoes count is increasing in the untreated area. In treated area, mosquito count is decreased. According to this result, approximately 38% of mosquitoes reduction was observed. By the end of the observation period, the treated area still had a higher mosquito count compared to the untreated area. These results indicate that water treatment is not effective in reducing mosquito presence over the observed period. Figure 2 shows the results of Osimum sanctum essential oil efficacy test for Aedes mosquitoes. In this test, here also blue color line is represent the treated part, red color line is represent the untreated part. Treated part had higher amount of mosquitoes, and untreated part there is none of any mosquitoes. Then during the observation period, mosquitoes count were decreased in the treated part. In untreated part, mosquitoes counts were increased. This reduction percentage is 64.52%. This figure 3 and figure 4 shows the experiment 2 and 3 trials of this Aedes mosquito repellent test. Here also mosquito reduction percentage is approximately 68 and 64%. According to these results suggest that Osimum sanctum essential oil could be a promising solution for mosquito control in providing a significant reduction in mosquito population compared to the untreated areas. These are the this image shows formulated mosquito repellent balms. A contain Osimum sanctum essential oil with 1%. B repellent balm is contain essential oil 1.5%. C is non-essential oil repellent balm. D is Osimum sanctum essential oil with beeswax and coconut oil mixed repellent balm. These are the conclusion. Finally, Osimum sanctum plant has a rich bioactive compounds like alkaloid, glycoside, tannin, flavonoids, and phenol in extracts, confirming its medicinal potential, and it is an effective mosquito repellent. It demonstrated significant mosquito population reduction varying 63% to 68% with essential oil, proving its effectiveness as a natural repellent. Then developed mosquito repellent balms combining essential oil, beeswax, glycerin, aloe vera gel, emulsifying oil and preservatives, and coconut oil for effective protection. These are the acknowledgement and references. Thank you very much.
Okay, now it's open for discussion. Any question from the audience? Any question you want to ask? Just one thing I want to know, that did you analyze the essential oil?
Yes, essential oil.
How did you do that?
TLC I did, and I did phytochemical test for methanol and aqueous extract.
How did you get the essential oil?
Essential oil hydrodistillation process.
Okay, then you have only the volatile.
Yes,
Then how?
100%.
Yeah, what method you use to see the chemical constituent in essential oil?
Essential oil content, GCMS.
Did you do that?
No.
Right. So do you know what major constituent?
Eugenol is a major constituent.
No, it's not eugenol. It's methyl eugenol, right?
Yes.
Not eugenol. Okay. And also, you know, in your research, you find right when you looked at the efficacy.
Yes.
This methodology, is it established one or you developed a method?
I developing the. I counted mosquitoes count, repelling mosquitoes count, and put in the machine and take.
No, I mean the method that you use to look at the repellent activity, that the
Yes.
box method.
Yes, ma'am. Yes.
How did is it you establish it or somebody else done and you use the same method?
No, I established.
You establish it. And how many replicates you have done?
3.
So it's not in your research. You have not given standard deviation or standard errors in your, right?
Yes.
Okay. Right. You.
Okay, then if you develop the method, then it's another research. Is there's no any such kind of research done before?
Kellen University, they are did for mosquito repellent cream they developed.
Okay, then there's this. You use the Aedes.
Aedes mosquito.
Is there any reason for use them, or is it effective for the other mosquitoes?
Now world facing dengue is major threat. That's why I use Aedes mosquitoes.
Okay, then when you present, I think it's better to say these things. Okay.
Okay.
Now this is not a competition or anything. We are giving you the ideas and other things to
Okay.
improve. Okay.
Okay.
And also these things that you mentioned in the conclusion, flavonoids and steroids, and these are the reason for the repellents. If you take any phytochemical, they have these, right? In the conclusion, you cannot say these are the reasons. You have to mention if you find that, as Madam asked, if you've done the GCMS or something, that compound is the reason.
Okay.
Otherwise, these are group of chemicals. We cannot say the conclusion, this is the reason. Right?
Okay.
Then if you take even some guava, pear, amber, mango tree, if you analyze, you will get the same results. You know that flavonoids are there, the steroids, all the things are there, right?
Yes.
Then we cannot use that. The reason is specified for this one for the repellents, right? Then you have to say, or you have to use the literature to support that.
Okay.
Okay, then thank you. Okay, nice work. Okay.
Thank you, sir.
Okay, we'll move to the next presentation. That is by Madhuanta Sisindu Kodikara. The title of the presentation is In Silico Investigation of Inhibition of TGF-beta1 Signaling Pathway by C-glycosidic elagatanin, a bioactive molecule from plant Obeschia ocandra, for their potential as a treatment option for hepatic fibrosis.
Good afternoon, everyone. I am Sasinda Kodikara, and I am here to present our research findings in silico investigation of inhibition of TGF-beta1 signaling pathway by C-glycosidic elagitanin, a bioactive molecule from the plant Osbekia octandra, for their potential as treatment option for hepatic fibrosis. Here's the outlines. So moving on to the introduction, liver fibrosis is a lethal disease caused by abnormal fiber formation of hepatocytes. This disease causes the activation TGF-beta1 pathway upon chronic stimulation by pro-inflammatory cytokines. Commercially available synthetic anti-fibrotic agents provide undesirable side effects over the potency of prolonged use. Therefore, natural medications are promoted over the synthetics by consumers. Thus, observation of potent natural compounds which can antagonize TGF-beta1 pathway receptors, which are TGF-beta1 receptor and TGF-beta2 receptor, is essential. In fact, selecting in silico drug development method for designing of advanced medications for liver fibrosis is more beneficial. So here's the research objectives. To investigate the in silico inhibition of TGF-beta1 pathway by C-glycosidic L-arginine, bioactive molecule of the Osbekia octandra plant based on binding interactions and binding energies. So considering to the methodology, we first obtaining the crystal structures of TGF-beta1 and beta2 via homology modeling by Swiss modeling. After that, binding site analysis was done by using Frank Web server. The 5 different L-argitannins, casuarinine, castelladine, vescalagine, and epiponicacotin A were prepared and geometrically optimized by using semi-empirical AM1 approach by the Gaussian software. On the other hand, selecting and geometrically optimizing commercial drugs, we done as the same, using same method. Silmitisertib drug used for TGF receptor 1 and ITD1 used for TGF-beta receptor 2. Then molecular docking part was done by using Autodoc Vina wizard by using Pyrex prescription. And finally, we analyzing the binding affinities, binding interactions with their base poses of each ligand toward each receptor, which respective drug towards each receptor by using Discovery Studio. Here's the result and discussion part. The TGF-beta receptor 1 and receptor 2 crystal structures were selected based on the highest GMQV value. So you can see the GMQV values of 2 different receptors, and they are also have the crystal structures of TGF-beta receptor and receptor 2. So when GMQV value above to 0.7, the complex is more stronger and more accurate. Here you can see the binding site analysis result we got from the PankWeb server. Here you can see the binding amino acid residues of TGF-beta receptor 1 and receptor 2 as the binding sites. So binding affinity values of 5 different L-argitannins you can see here, and with the commercial drugs towards TGF-beta receptor 1 and receptor 2. So among 5 different L-argitannins, casuarinine shows the highest binding affinity value when comparing to commercial drugs towards TGF-beta receptor 1 and receptor 2. So here you can see the drug binding interactions of TGF-beta receptor 1 and 2. And here you can see the casuarinine binding interactions of TGF-beta receptor 1 and receptor 2. So here you can see in dark green color, strong hydrogen bonds are shown, as well as their reference casuarinine drug, you can see the same bonds as well. That means the activity of the binding interactions and the referent drugs also same, and they give the same activity. So conclusions, among 5 different L-argitannins, casuarinine showed a high and comparable binding affinity towards TGF-beta receptor comparing to commercial drugs. Thus, casuarinin containing novel medication is recommended to be formulated as an anti-fibrotic medicine. So I would like to express my sincere gratitude to Dr. Rangesh Jayakodi, Anushanga Rodrigo, and Mrs. Shehara Gunawardena to give me the moral support. And finally, I would like to thank IPRC 2024 University of Kelaniya arranging this platform to give me this opportunity. Here's the references. And thank you for listening.
Okay, thank you, Mr. Kodikara. Now it's open for question for the audience. If you have any questions, you can ask. Okay, Mr. Kodikara, it's nice work you have done. The one thing I want to clarify that is this done actual research that is done? These are inhibiting TGF-beta 1 is still yes or no? That means, have they done the actual research?
Yes, we done the actual research.
That means not the computational in the labs. They have done this one.
Computational wise, using
computational wise, not the real.
Using softwares.
Softwares. Oh,
yeah.
Softwares only. Okay.
Softwares.
That means next step is to do somebody to do
formulation development
and actually do the experiments, right?
Yeah. Yes.
Okay. That you are taking that the least binding energy is the good one, right?
Yeah.
Is that the true for the all the time?
Here, yes, we can predict that from that that software when the GMQE value stands for global model quality estimation, so that we can predict it's stable.
Okay, that means this go and bind into a binding site somewhere?
Yeah, binding sites.
Those are the amino acids that you mentioned, right?
Yes.
Is that similar for the commercial drug?
Yes, similar for the commercial drug.
Is a commercial that is you got the reference or you did the commercial drug also? That
no, actually we got that reference drug from the literature.
And compare this.
Yes.
Okay.
Those are commercially now available at the market, that drugs. So we compared with those.
Okay, then thank you.
Thank you.
So what is the common name of this plant?
Heme bovitia.
Heme bovitia. Yeah, it's well known for fatty liver, right?
Yeah. Yes.
So whenever you do studies, did you check any references? What type of compound? There are similar compounds affecting the both. You have any idea? Because there are. I know that using this plant, the extracts or the the powders are developed as the capsule or pills, so they are available in the market, and it says very effective. I was just thinking, did you do any literature?
Yes, actually we did, and there's in heme bovitia plant there are 5 different L-arginines. So the casuarinin is taking the antagonist activity to the liver fibrosis.
Okay.
So that we focus.
Okay. Thank you very much. Okay.
Thank you.
Okay, thank you, Mr. Kodikara. Our next presenter is Shanaka Karunatilaka. He's going to present about Paspangu herbal remedy potentiates antibacterial efficacy of penicillin against methicillin as resistant Staphylococcus aureus.
Imaging a world where our most trusted antibiotics can no longer protect us against deadly infections. Hospitals around the world, this is already a reality. Good afternoon to you all. I'm Shana Karunatilaka from University of Ruhuna. Today I'm excited to present our study titled Paspangu Herbal Remedy Potentiates antibacterial efficacy of penicillin against methicillin-resistant Staphylococcus aureus, or MRSA. These are the content of my presentation. So when talk about the antibiotic resistance is a critical global problem because of there are some resistant bacterial infections very difficult to treat because of there are multiple antibiotic resistance, thereby increasing mortality rate and also greater the economic burden and economic burden. So in Sri Lanka, there are some traditional remedies to treat common illnesses such as cold and fever. So in Paspangua is a type of traditional remedy, and people believe it has potential to combat against multiple resistance, and also it contains blend of 5 or more herbs that are believed to have therapeutic properties. So with limited options to counteract these resistant bacterial infections, so our study investigate that whether this Paspangua Ayurvedic formulation could provide new approach to that resistant bacterial infections. So here are our main objectives. Those are to investigate the interactions between Paspangua formulation and penicillin, and to identify specific plant species within Paspangua that impact antibacterial activity. So let's talk about the methodology part. So first we collected Paspangua Ayurvedic formulation that are available in market. Then actually we collected 5 commercially available Paspangua Ayurvedic formulation, and we prepared according to the label guidelines. So then we checked the antibacterial activity. We follow the well diffusion method. So we use 50 microliter of 3 milligram per milliliter concentrated positive control, and also distilled water as a negative control. So then we checked and we recorded inhibition zones. Then as a next step, we check penicillin and Paspangua extract were combined and assess their interactions. So here we use 25 microliter of penicillin and 25 microliter of each and every Paspangua decoctions separately. So here we use half drug amount compared to the positive control because we use 50 microliter 3 milligram per milliliter concentrated penicillin as a positive control, and also 25 microliter 3 milligram per milliliter concentrated penicillin, and also 25 microliter distilled water. So as a next step, we investigate what is the responsible plant that enhance the antibacterial activity against MRSA against penicillin, against MRSA. So here we collected Tippili, coriander seeds, Aratta, Vishnukranti, Patpadagam, ginger rhizome, black pepper, Katuval Batu, and Venival Getter. Then we use 25 grams of dried individual plants material. Then we boil with 200 milliliter distilled water in 20 minutes. As a previous method, we followed the preparation of Paspangua Ayurvedic formulation. Then we use 50 microliter of 3 milligram we also use the antibacterial well diffusion method, and the diameters of inhibition zones were recorded. So all the inhibition zones analyzing using one-way ANOVA followed by Dana's post-hoc text, and also data were analyzed using SPSS 20 software. So we'll talk about the interesting part. So different Ayurvedic formulation of Paspangua Ayurvedic formulation does not show any inhibition zones against MRSA only, but in combination with penicillin, compared to the positive control, each and every Paspangua Ayurvedic formulation show the antibacterial activity against MRSA. And as a next step, when talk about the responsible plant that are including in Paspangua Ayurvedic formulation, compared to the positive controls, coriander and the Pipernigram plants are significantly enhanced antibacterial activity compared to the controls. Here we use penicillin, 3 milligram per milliliter concentrated penicillin, 25 microliter with distilled water. So compared to that control, the significantly coriandrum and Pipernigrams are increasing their antibacterial activity against MRSA. So when talk about their MIC value reduction, you can see the MIC values are gradually reduced. So initially, the MIC value of the penicillin is 17.52 microgram per milliliter. So when talk about the combinations like coriandrum and the pipernicum, so when talk about the coriandrum and the penicillin combinations, we can reduce the MIC values of the drug for 4.17 microgram per milliliter, and also when using the pipernicum, we can reduce the MIC value of the penicillin into the 9.88 microgram per milliliters. And also you can see the percentage value of the drug enhancement against MRSA. When using the pipernicum, you can see 43.79% and also 76.28% when you are using coriandrum seeds. So as a conclusion, traditional herbal formulations like paspango could serve as adjuncts to antibiotics, and also particularly creating the resistant strains like MRSA. And also here, coriandrum sativum and also pipernicum, in Sinhala we call kottamalli and gummiris, so are the primary potentials in the enhancing penicillin antibacterial efficacy. And also, this study highlights the importance of exploring polyherbal interactions to develop alternative strategies against resistant infections. And coriandrum sativum showed potential activity enhancement with penicillin, offering a pathway to reduce antibiotic usage and resistant development. And also, plant metabolites may interact with bacterial cell wall or inhibit the MRSA defense mechanisms, enhancing penicillin's effectiveness. And our next, as future recommendation, we recommend to isolate the bioactive compound that are responsible for that the drug enhancement, and we are using NMR-like techniques and elucidate the structures and develop the new formulations. So these are some of the references that I used to prepare this presentation, and thank you.
Okay, thank you, Mr. Karnatilaka. Now it's open for questions. Okay, if not, okay, why you got this idea to mix this one?
Actually, paspango Ayurvedic formulation is widely available in Sri Lanka, and most of the people, at least one time, they consume their life. So, and most of the people believe that it has therapeutic properties. So that's why we select that paspango Ayurvedic formulations, and we hope using that the blend of 5 or more herbs, and it's better to understand. And actually, the penicillin is not available in a market because of the resistant development. So few years back, the penicillin is served thousand lives. So we can reintroduce penicillin by using that combinations.
Okay, that you have done with the individual compounds that you did with like the pepper and other things, right?
Yes.
In that one, did you make the mixture by yourself?
Yes, actually, separately collected each and every plants that are in paspango formulations. We collected 9 plants, then separately collected the plants and checked. Yes.
Then it is not paspango.
Yes, even though it mentioned as paspango, there are some paspango formulation contain 10 plant species, and
then
some are 6.
Jasapango, right? Deviations. Okay. Then you did each
plant collected
and you used the commercial ones.
Yes.
Then the commercial ones, they didn't work like that, right? According to yours, some of the commercial ones, I heard that they are using adulterants, like adding compounds, right? But in your one, it says it's not. They are not.
Actually, sir, when analyzed the labels, and we saw that the different plant species they mentioned, then we collected several plant species, and using herbarium shades, and we identified in Pinnaduwa Medicinal Garden. Then after confirmation process, we start our works.
Okay. Then are you planning to do some other bacteria too? That.
Yes. Actually, we focus mainly about penicillin is a type of drug that are widely not available in market with the resistant development. It's better to use the different resistant bacteria and expand the procedures and mechanisms.
Okay, thank you.
Now, what you say is right, that even though it says paspango, I know that there's more than 5 ingredients. Whenever you collect samples from the market, did you find common ingredient in all of them?
Yes, actually, Pipernigram, Coriandrum sativum, and Aratta, and Syngiber officinalis and type of plants are commonly available in different Paspango formulations.
Is it because they are adding other plants to improve the efficacy?
It may be they added to enhance the flavor.
You
and
didn't
smell.
check the efficacy, right?
No, just
your
to check
purpose is different,
yes.
right? Oh, okay. So, and also like, whenever you, you just collect them from the sample that are available in the market, then did you get the extract of that?
Yes, we collected 20.
That's you prepare the decoction.
And then we didn't concentrate it because of we don't need any prescription or any supervision under.
You didn't do freeze drying or anything.
No,
right?
we didn't use because
so the concentration you don't know.
Actually, we used 25 grams and boiled with 200 milliliter of water. Then we take that concentration.
It's good that if you could freeze dry and take the the extract and at least weight by volume.
Yes,
if you could have,
yes, madam. But the thing is, madam, people are consuming with other drugs, Paspango, without any prescription. So it's better to.
And also the other thing that I want to know that according to the Ayurveda, the text, what they have mentioned about Paspango, they must have defined it, right?
Yeah.
Did you check that?
I checked, madam, but different. Most of the time we couldn't find any specific terms because they don't like to share the things.
Okay, thank you.
Thank you.
Okay, thank you, Mr. Shanaka. Our next presenter is Sandarage Manupurabat Kaumadi Premachandra. Her title is Development of Herbal Tea to Manage Gastritis and Evaluation of Acid Neutralizing Capacity.
Good afternoon, everyone. I am Kaumadi Premachandra, an undergraduate student of CINEC campus, Malebe. Our research topic is development of herbal tea to manage gastritis and evaluation of acid neutralizing capacity. Our research group members are SMK Premachandra, WADJ Tanishka, and TMSA Tennakon. Our research group supervisors are PM Kumara Peruma and SLA Gunavardhana. These are our research outlines. Have you ever suffer from gastritis? Most of the time, the answer is yes. Gastritis is one of the most prevalent medical conditions among the world population. There are two types of gastritis, acute gastritis and chronic gastritis. There are herbal teas are used to treat gastritis in various disease conditions. So, we are interested to making a herbal tea bag to manage gastritis. There are many herbal teas are used to treat gastritis. Upon that, Aegil marmelis, Desmodium triflorum and Centella asiatica are leading ones. Aegil marmelis is known as belly in Sinhala. It is also known as bale. It has antibacterial and gastroprotective activities. Desmodium triflorum, also known as Undupialia, it has anti-inflammatory and analgesic activities. Centella asiatica, also known as gotukola, it have anti-ulcer and anti-tumor activities. So, these are our research objectives. The general objective is to develop a herbal tea bag using Aigil marmelis, Desmodium triflorum and Centella asiatica in different ratios to manage gastritis. Another objective is to evaluate the acid neutralizing capacity and duration of consistent to understand potential therapeutic activity of the herbal tea bag. Here is our methodology. Firstly, we use Centella asiatica, Aigil marmelis and Desmodium triflora separately. Then we shade dry them. Then we got these dry powdered and make 2 grams tea bags. Then we use these tea bags to evaluate the acid neutralizing capacity in 2 conditions. First condition is the prepared tea bags were infused in 100 Celsius of constant temperature for 357 minutes. In there, we evaluated the which tea bags given the best acid neutralizing capacity and the best performing time. According to the data, the best performing time is 5 minutes. Then, under the second condition, 5 minutes constant time applied. The tea bags infused for temperature difference. Then we applied 100 Celsius, 80 Celsius and 60 Celsius for 5 minutes constant time. There we evaluated 100 Celsius tea bag as the tea bag is best performing one. We used green tea as the positive control and negative control as the distilled water. Then we used these tea bags for evaluation of duration of consistence. In there, this picture shows the modified model of Vertier's artificial stomach. We use this model to evaluate the duration of consistence. In this, we use positive control as the green tea and negative control as the distilled water. Here is the results part. In this, the first figure shows in vitro acid neutralizing capacity at the 100 Celsius for 5 minutes. In there, T9 test sample value is 24.70 milliliters and the T7 test sample is 27.10 milliliters. The green tea value is 5.43 milliliters and distilled water is 0.73 milliliters. The highest acid neutralizing capacity was observed by T9 and T7 samples. Then the second figure shows the duration of consistent neutralizing effects on artificial gastric acid. In there, the T9 sample, which contains Aigil marmelis, showed the value 382.33 seconds and the green tea consuming 42.00 seconds and when consuming distilled water 72.00 seconds were recorded. Then we concluded the study showed that the T9 formula, which contains only Aigil marmelis, is more effective in neutralizing artificial gastric acid compared to other plant combinations. The best results for treating gastritis were found when the plant were infused at 100 Celsius for 5 minutes. I gratitude to our supervisors, Ms. Pavitra Kumarak Peruma and Ms. Shahara Gunawardana for their guidance and encouragement. And special thanks to our faculty dean and the lecturers of the Faculty of Health Sciences, Sinek campus. These are some references we used for our research project. Thank you.
Okay, thank you, Ms. Premachandra. Now it's open for the questions. As usually, no questions. Now, before go to the experiments, I want to know what is that acute and chronic gastritis?
The types of gastritis types.
What it means. Yeah.
Chronic means some a long go one. Acute can be another.
It's like 1 day, 2 day, like if you
Sorry, sir.
sudden gastritis, that called the acute one.
Yes, the chronic one is a long period time one.
That is developing with the time, right? Okay. And your experiment, can you go to that slide that you mentioned that 100 Celsius, there's something, this one. Now you that one for the 100 Celsius and you did 357 minutes and
Yes,
you choose 5 minutes is the best.
Yes, sir.
And then you use the 5 minutes to check the 190 and 60 Celsius.
Yes.
Then you got the best one is 100 Celsius.
No, sir. Second one we use 100 Celsius, 60 Celsius, and 80 Celsius.
Okay.
Yes.
Then you use the 5 minutes.
Yes.
Right? Then you found that 100 Celsius is the
Best one.
then obviously that is true because you have done the 100 Celsius and you found the 5 minutes is best. And you are doing this 190 and you put here 90, okay? 80 and 60. Yeah, then you are doing for the 5 minutes. Obviously, the 100 that is optimized for the 100 Celsius, right? In the 90 and 60, 80 and 60, it would be more time because you know I mean.
Yes, sir.
You did something and you are testing other way around.
Yeah.
You have to. You
Yes,
got
sir,
what I mean. This is not a criticism, but that is the experimental error I see there. Right? Okay, that's fine. And then that's how we learn. And then you haven't done any statistical analysis, have you done?
Yes.
But when you go to your graphs, we don't see that. That means error bars and other things, right? You have to mention
We
this.
don't edit, sir.
Okay. And the other thing is that method, neutralizing assay, is that the method that you found or it's already there?
It's using the Fordran method, acid
Okay,
neutralization.
it's already using method,
Yes.
right? Okay, that's why for all the others also, now the new method of putting the reference, we are putting under the slide, the particular slide. Then we are clear that is from a reference. Then we don't know, now previously there's assay, she found it, and then we know it's she found. And if you put a reference, we know that it is, you are following that reference and you are doing that, okay?
Yes.
Okay, thank you, okay?
Thank you, sir.
Okay, then thank you very much and nice presentation. And these are two constructive comments, okay? Don't take it for the personal, but you are learning these things, right?
Yes.
Okay.
Thank you, sir.
Then next presenter is WMRG Varuna Veerasekara. His title is Preparation and Bioactivity of Indigenous Formula are used as hair care oil. Good.
Good evening, distinguished guests and everyone. Today I came to speak about, present my research work under the topic of preparation and bioactivity of indigenous formula used as hair care oil. So this is the outline which I am going to talk about. As introduction, I can say as humans, we all concerned about our appearance. So the hair care is a vital part of the day-to-day routine. So under that, when we talk about the hair care, there are so many aspects. From that aspect, hair dyeing is a crucial aspect. So when talking about the hair dyeing, there are so many products available in the market in many types as a powder form, semi-solid form, and oil. So, and the other thing is those products can be synthetic or natural. So in the ancient time, with my findings, I have found that most of the products are natural products, but due to the high demand, due to cater the market, now many synthetic products have evolved to the world. But by the long-term use of those synthetic products, there may be some effects for our health, like hair damage, skin irritations, allergies, cancers, and even death. Because there may be some colorants, chemicals, peroxides, and preservatives using to produce to make the synthetic products for their the convenience for the manufacturers and to get more profits. So for that, as a contribution to transfer the demand to synthetic products to the natural products, I have formulated and evaluated dyeing hair care oil using natural, herbal, and authentic materials instead of synthetic products, which can be used for long term to minimize the health effects to the human well-being. So for that, I have used Kikirindia plant, sesame oil, aerial roots of Vatakeya, Pandanus tectorius, and Tripala, Katukarandu, and Adamata plant, and other than that, the iron powder. With reference, these ingredients were taken with reference to Ayurvedic resource named Thalpate Piliam. And with these ingredients, I formulated the oil. So first, the decoction was made. Decoction was made from the plant parts, and the juice was made by the Kikirindia. Only from Kikirindia the juice was made. Then the decoction and the juice mixture was mixed and heated till it makes 1/4 of the original solution. So then the decoction and the juice mixture was filtered, and then another part was done from the sesame oil. The sesame oil was also heated, and then the iron powder was added. So after that, the iron powder added sesame oil was filtered. Then the decoction and the juice mixture, and the filtration of the iron powder and sesame oil was mixed and again heated. Then after coming to the room temperature, it was filtered, and after that it was sealed in a copper pot using clay, and one sample was kept underground, and the other sample was kept in the normal environmental conditions as per a control sample. So after that, the sample was named as, after a month, the underground sample and the normal condition sample was taken out and removed the seals. And the underground sample was named as sample A, and the normal environmental condition sample was named as sample B. So after that, this was evaluated in many aspects, like organoleptic properties, physical properties, chemical properties, stability, bioactivity properties, and heavy metal analysis. Under the organoleptic properties, the color, odor, and appearance was tested. Under the physical properties, pH value, moisture, sedimentation, some other things was tested. And the chemical properties, acid value, peroxide value, was tested, and the iodine value was tested. For the stability, only the organoleptic properties was done upon 3 months time by taking equal samples of the final product, keeping in the normal one sample in the normal condition, one sample in the refrigerator, and one sample exposed to sunlight. So then the bioactivities was done as an antioxidant and anti-inflammatory. So then other than that, the heavy metal analysis was done. So as you see, the sample A and the sample B was taken separated into a new containers. So then the heavy metal analysis was done using ICP-MS by the digestion method. So bioactivity was done from hexane, dichloromethane, and methanol. So in the extraction, the methanol extraction was didn't got the methanol extraction. The highest extraction was taken from came from N-hexane. So the sample AH, sample BH is named as the H is coming to the N-hexane. The sample AD, AB, the D is for dichloromethane, and the sample AM and BM, the M is for methanol. So for the heavy metal analysis, lot of heavy metals was analyzed and get only some the lead was there, and in only in one sample. Then these are the bioactivity results. This is the antioxidant result for the samples from dichloromethane, which is here the standard was the BHT, its value is 26 ppm, sample AD, which is on the underground sample 223 ppm, and the sample from B, dichloromethane, extracted from dichloromethane is 34 ppm. So then this is the antioxidant results for the samples extracted from the hexane, and this is the results for anti-inflammatory tests taken. Here the standard was taken as aspirin, and these are the IC50 values. So so then other than that, I have to tell about the results of these also. So from when we talk about the organoleptic properties, the color was dark blue color, and the odor, it was a characteristic odor, which and with that when we talk about the stability, the color, odor, and the sedimentation properties have not changed. Only the sedimentation test, there were some changes in the sedimentation, but not a lot. And the color and the odor has was not changed. So the physical properties, the pH was somewhat acidic, and the moisture, there was some moisture content in the sample when doing the test. And the chemical properties, the acid value, there was the small low acidic value, and somewhat high iodine value, which makes it a good quality. And so yeah, those are the results. So as a conclusion I made, I can say this is if we can produce the natural products like this, is good for our health, for the living beings. So for when we consider about this product, it's good to use for humans, and but have to do some further tests, further analyze, and clinical tests also, and can commercialize this product in the future. So I would like to thank Professor Priyani Paranagama for the guidance from the initial point to the last point, Professor Molligoda, Professor Harapaddeniya, Institute of Chemistry, Ceylon, and Department of Chemistry, University of Kelaniya. And thank you.
Okay, thank you, Mr. Veerasekara. Now, is this a hair dyeing one, or hair to use to dye your hair, or to protect your hair?
To dye the hair. The main focus is, the main intention is to dye the hair.
Okay. Then the hair care oil means that
There are some other properties where we try to include some properties because there are some more properties are coming from the natural plants that we are used. So we can, with the literature, have shown there are some more properties from sesame oil, Kirindia plants and all things. So they are, by the other hand, they are giving the protection to the hair also. Main intention is for hair dyeing oil.
Hair dye.
Yes.
Is it dyeing in the blue color or black color?
It's actually black color, dark black color. Actually, I couldn't say that I have done some trials with hairs taken from some volunteers, the dry hair and the black hair, and did some trials upon the research time. So I have applied this hair dyeing oil and then synthetic oil and washed after some time. But there were some darkness came, but with the developments, I have to develop the product.
Yeah, I asked question that because you said there's a blue color one that at the end it's become black.
It's a dark blue color. The oil is dark blue color.
Dark blue going to be okay. And because nowadays not everybody doing the black, they are purple and multicolor.
But according to the literature in the resource in the Ayurvedic book, it says can be developed to make the color blue in the hair.
Is this a formula that you made or is it already written in the Ayurvedic books?
The resource book, the names of the recipes was there, but we have to we have to do the analysis what amounts which have taken to this to formulate.
Okay.
From these plants.
Okay, then in the Ayurveda they use the iron powder.
Sorry.
You said iron powder.
Yeah, iron powder also.
In the Ayurveda.
Actually in the recipe it was mentioned to use iron powder taken from irons.
Yeah, then in your ICPMS one, I expected more iron than what you had because you are already adding that.
Yeah, adding, but filtering it finally.
I think iron is you are using to absorb some stuff, maybe, right?
Actually,
maybe the toxic chemical and other things.
The iron powder is giving another task, doing another task by strengthening the product and giving somewhat effect to the black color also.
Okay. Okay, then the only other thing that the one you showed is a one commercial one you showed. Is that a real one or is the commercially one that?
Sorry.
That bottles, 6 bottles or something with the name.
I showed that transferring from synthetic ones to natural ones.
Okay. Because you will get some court cases if you put those. Okay, then thank you very much. And are you using that oil? Do you
Not yet. Have to.
Because you have a nice hair, that's why.
Have to develop by doing clinical trials. We cannot do the clinical trials up to the stage. I have to get approvals to do the clinical trials and all things.
Okay then. Thank you very much and nice work. Okay, then we are moving to our last session, last talk. It's from Siddharth Ravindra Lianage. He's going to talk about, is he present here? Mr. Siddharth Ravindra Lianage. Social media ancestry detection using prompt engineering techniques. Is absent. Did you call him or inform him? Okay then. Okay, we put that. Okay, then, madam, can you come? No, madam, do that. Okay, one thing that I realized now, these are most of the things from the Ayurveda. That's why we are chemists. We are so much onto those analyzing things, and that's why some of the things are missing. And our madam, that Professor Parnagama, is working too much on this field to these Ayurvedic things to get these things to the commercialize. Otherwise, if we are not giving this evidence in the scientific way, we cannot go for the higher market. That we are having the group products, right? But we cannot go. That's what, madam, that we are doing this, right? And she has done a good job and is still doing. And she also our mentor and my teacher, and then still they are working very hard, even than us, I think. Okay, then, madam, can you conclude, give some words?
Okay, now this is our last session and of the 24th International Postgraduate Research Conference, IPRC 2024. I think there were 6 presenters. So as Dr. Kadigamu said, that most of them related to indigenous medicine, and what we wanted is to upgrade the the standard of indigenous medicine available in Sri Lanka, mainly because we have a lot of resources and many medicinal plants are available, and so this indigenous medicine is well accepted by other countries. So we wanted to get the global recognition. So therefore we are happy that students are involved in doing research on that. So with that remark, I would like to conclude this session and congratulating all the presenters. Thank you very much for submitting your the abstract and presenting here. So we wish you all the best, and so with that we'll conclude this session. Thank you again.
Thank you, everyone. Now I would like to invite the chairs of the session to award the certificates to the presenters. Please come forward to receive your certificates. I would like to invite Professor Priyani Paranagama to give out the certificates. K.A. Aruna Indrachapa Alvis. Muhammad Haris Fatuma Rihana. Madhuwanta Sasindu Kodigara. Now I would like to invite Dr. Chamila Khadigamur to give out the certificates. Shanaka Karunatilaka. Sandarage Manuprabha Kaumadi Premachandra. WMRG Varuna Vishwajit Veerasekar. Thank you, sir. We would like to convey our thanks to the presenting authors for their submissions and the many interesting concepts, ideas, and applications that were set out on different areas. We trust that based on your findings presented here and the discussion, you will be able to improve and further refine your research study. On behalf of the organizing team, we would like to thank our conference chairs, Professor Priyani Parnagamage and Dr. Chamila Khadigamur, for providing their professional input to the authors and the audience here, and hope that the discussion will help the authors to refine their work further. Thank you, Professor Priyani Parnagama and Dr. Chamila Khadigamur, for your presence today. As a token of appreciation for the hard work and dedication, we have arranged a small gift for you. Please come forward to receive it. Thank you, ladies and gentlemen, for your presence here today. That concludes this session of the 24th International Postgraduate Research Conference 2024. Thank you for participating. Have a nice evening.